So welcome back, everybody. I hope the next speakers are available. Vicky, are you guys available?
Yeah. I think I'm.
Okay, perfect. Yeah, you're there. It's there. Sorry. So, starting the next session, we start with a presentation by Yenjun Lee. Is it?
Perfect.
Excellent. Called the synthetic nervous system controls the biomechanical model of aplysia feeding. So please, when you're ready, take it away.
Okay, so yeah, let me try to share my screen here. Could you see my slides now?
Yes, we can.
Cool. Okay, so yeah, let's start. And thank you all for coming. Today I'll talk about my research on a synthetic nervous system controls a biomechanical model of aplysia feeding. So I'll first introduce the motivation of our research. For animals, a critical capability for their survival in a changing environment is generating multifunctional control. For example, frogs can use their legs to exhibit swimming, walking, and hopping. And their ants can use their mandibles to cut stalks of grasses and move them to their nests. Once in their nests, they use their mandibles to manipulate the grass. And the sea slug Aplysia californica is a tractable animal to study how animals can generate multifunctional control because it has a relatively small nerve system. But it can demonstrate multifunctional behavior during feeding, including biting, swallowing, and rejection. And our research goal here is to understand how aplysia can generate such multifunctional feeding control. And to meet this goal, we need a computational model framework with predictive abilities, and then we can use it as a test bed so that, for example, we can test our hypothesis using it. And there have been some research focusing on modeling the neurodynamics of aplysia feeding, but seeing these researchers' the interaction between the neurodynamics and the biomechanics are often ignored, and those models ignore these interactions generally have limited predictive abilities. Our solution here is use a neuromechanical model of feeding behavior in Aplysia californica, and it emphasizes the feedback and the biomechanics so that the model can generate multifunctional behaviors and adapt to the external loads, just like we can see in animals. So I will first talk about our biomechanical model, then present the neural control model. And the last part of my talk is about the multifunctional feeding behaviors generated by the model. Okay, so the first of all, we build the biomechanical model represents the feeding apparatus of aplysia. The model consists of two segments, a head and a grasper. Here we exaggerate the length of the embedded buccal mass a little bit so that we can see the protraction and retraction of the grasper more clearly. The real shape of the buccal mass is more round. And we model the connection between the head and body as a prismatic joint with a single degree freedom. There is another prismatic joint connecting the grasper and the head. So the biomechanical model here has 2 degrees freedom in total. And we also model several key muscle groups that contribute to the feeding behaviors, like I2, I3, I4, and the hinge muscles. And in our model, their dynamics are abstracted as second-order systems. And the biomechanical system can also interact with the environment. So generally in our model, the environment is a seaweed segment fixed to a force transducer. And the biomechanical model can interact with the seaweed via 2 contact points. The first contact point is between the grasper and the seaweed, while the second contact point is located at the jaw. And the normal contact force is determined by the activation of I3 and I4 muscles. Then the tangential contact force acts like the column friction force. So given that we have biomechanical model, we start building the neural control model that reflects the neural dynamics and the neural circuitry critical for application feeding control. So we use synthetic nervous systems, or SNS, to represent neural dynamics in our model. And the SNS are relatively simple, but still biologically plausible models of neural systems. Here is the schematic of the SNS. And the SNS is a conductance-based model, which means that it models important conductance components, including the chemical synapses, electrical synapses, and some other important ion channels. And generally, the SNS treat a neuron as a single compartment model, and by neglecting the intracellular resistance. Furthermore, the SNS we use here is a rate model. So instead of generating spikes, it uses an activation function to represent the relationship between the membrane potential and the neural activity. So here on the left, we can see the difference between a spiking model like Hodgkin-Huxley model and the rate model like SNS. And one advantage of using SNS is that it balances the biological plausibility and the computational complexity. As we can see on the right, the simulations of SNS is faster than Hodgkin-Huxley model. And using SNS framework, we developed a neural circuitry model of applicit feeding control based on non-synaptic connections, where we also take into account the feedback signals from the peripheral biomechanics. We organize the neurons into 3 layers, and the bottom layer is the motor control layer. It consists of 5 non-motor neurons innervating the key muscles we introduced before. Some of them receive proprioceptive feedback to regulate their activation, and the majority of them receive descending commands from the higher layer, which is the buccal ganglion layer. And in this layer, we organize the 9 buccal interneurons into 5 subnetworks based on their non-functional roles in biting, swallowing, and rejection. And the subnetwork can accomplish its function by stimulating or inhibiting a specific subset of the motor neurons. They also receive feedback signals so that the duration of the protraction or retraction of the grasper can be changed, enabling feeding to adapt to external loads. And on the right, we can see the detailed synaptic connections in the subnetworks. We modeled these, we determined these connections and the corresponding parameters in their SNS model based on existing literature. So if we can find non-parameters, we use them. If for those unknown parameters, we estimate their parameter values at our best. And the top layer is the cerebral ganglion layer. It contains common like interneurons, CBI2, CBI3, and CBI4, whose activation patterns encode which feeding behavior to generate. And the activation of these CBI neurons are determined by the feedback pathways so that they can coordinate the behavioral switching based on exteroceptive stimuli. And all these pathways and their synaptic connections can be found in existing literature. And to validate the model, we performed a simulation experiment to see whether it can generate multifunctional feeding behaviors similar with animal responses. And then my co-author, Dr. Gill, tested the behavioral and neural responses in intact aplysia to well-defined mechanical stimuli that elicited different feeding behaviors. And in our simulation experiment, we tried to adopt similar experimental setup so that the comparison between the animal experiment and simulation experiment is fair. For example, in animal experiment, to elicit bites, which are failed attempts to grasp food, the dried seaweed was attached to specific zones of aplysia until we can see the protractions of the grasper. And similarly, in our simulation experiment, we removed the seaweed after the onset of biting. For the loaded swallows aplysias in animal experiments fed on strips of unbreakable seaweed. And in the simulation experiment, we allow aplysia model to interact with the fixed seaweed segment. For the rejection behavior, the animal in the experiment rejected the tube by pushing it out of its mouth using its grasper. And in our simulation environment, we allow the grasper segment to interact with the movable tube segment. So we find the model can qualitatively generate multifunctional feeding behaviors, including biting, swallowing, and the rejection of Aplysia californica. Like intact animals, the neuromechanical model generates fast biting behaviors with similar protraction duration and retraction duration. The X sub GH here is displacement between the grasper segment and the head segment. The force is the force applied to the seaweed by the grasper. And here is an animation showing the movement of the head segment and the grasper segment of our biomechanical model. The rotation of the grasper here is added for illustration purpose only. So it's in our real biomechanical model, there's this degree of freedom hasn't been added yet. Okay, so the model can also generate swallowing. The protraction duration of swallowing in simulation is slightly shorter than the retraction duration. So it's similar with what we can observe in animal experiments. And there is a large positive force exerted on the seaweed during the retraction phase, indicating that the grasper is trying to pull the fixed seaweed into its mouth during this phase. And the model can also successfully generate rejection-like behaviors. The period of rejection behavior is longer than those ingestive behaviors. And there is a negative force during the protraction phase, which means that the grasper is trying to push the seaweed out during the protraction. And again, it is similar with what we can see in animal experiment. We also quantitatively compare the model behaviors with reported Aplysia experimental data. And from this figure, we can see both intact animals and our neuromechanical model can generate the biting with similar duration. This duration is relatively short, and the protraction phase and the retraction phase are basically the same. In contrast, the behavioral duration greatly increased if we study the isolated ganglion of animal where the feedback pathways are removed. This increase in the duration occurs mainly in the protraction phase, and with the less increase in the retraction duration. We also removed the proprioceptive feedback in our neuromechanical model, and we observed similar variation in protraction duration. The second dataset compares swallowing duration under different load conditions. When the animals feed on unloaded seaweed, the total cycle is around 5.5 seconds. And when they attempt to ingestive unbreakable seaweed, then the addition of the load actually slows down the behavior and increase the total duration by about 30%. In simulation, the unloaded seaweed can be implemented by removing the connection between the seaweed and the ground, and we can see that the output of the model follows the trend of animal dataset here. So to sum up, we presented a neural control model of epilepsy feeding based on synthetic nervous system, and it can control the biomechanical model of epilepsy in simulation. We found this neuromechanical model is qualitatively sufficient to generate 3 key different feeding behaviors. And by comparing the model's output with animal data, we demonstrate that the model has some predictive ability. In the future, we will compare the model responses with more animal data to further validate and improve our model. We also perform parameter sensitivity analysis to make sure the model is robust to variations in parameters. And we'll also generalize the model for robotic control. Our collaborators from Carnegie Mellon University are developing the epilepsy-inspired robotic grasper, and the neural control model presented here has the potential to realize multifunctional control of the robotic grasper. And I want to acknowledge the causes of the present work, as well as the responses found in my research. Thank you for listening, and I'll be very happy to answer your questions.
Great, thank you very much. So do we have any questions from the audience? And while you think I have 1 or 2 questions. Okay, Nicholas, Nick, would you like to go first?
Yeah, sure. Yanjun, that was an excellent talk. I really like seeing this model. Thank you for sharing that. So from what I understand previously, there was a Boolean-based control network that had similar multifunctionality, but now you've added all of these neural components on top of that, which I think is really great. I'm sure that adds some more biological dynamics and realism. So were there noticeable differences or benefits to including leaky integrator dynamics and conductance-based interactions throughout the network over the Boolean model?
Yeah, thank you for your question, Nick. Yeah, we have some discussion about that. The Boolean model is also very, it can achieve the multifunctional control. But in terms of the biological plausibility, it's like some, those biological components, and the parameters in the Boolean model are some kind of abstract. There's some threshold or something. Yeah, if we can tune those parameters correctly, it can also become, achieve those, for example, biting, swallowing, and the rejection. But here we use another way. Yeah, I think the comparison between our model and the Boolean model can be a very interesting part. Yeah.
Great. I look forward to hearing more about that in the future. Thank you.
Thank you.
Okay, for interest of time, we can continue some of these questions either on the chat or on Discord, so that we need to continue with the next presenter. So thank you very much, Yanjun. Very, very interesting
Thank you.
talk.
Let me end my
questions. So the next paper is Scaling a Hippocampus Model with GPU Parallelization and Test-Driven Refactoring, presented by and submitted by Jack Stevenson and Charles Fox. So if you're ready.
Okay, testing, 1, 2, 3.
Yes, we can hear you. So the
Lovely,
first,
and
yes.
you should be able to see the screen.
Yes, we can.
Lovely, we'll move that over there. All right then, let's go. So welcome to my talk on scaling a hippocampus model with GPU parallelization and test-driven refactoring. I'm Jack Stevenson, from a PhD student at the University of Lincoln, and we do have our own ePrint repository where you can have a look at this paper if you would like. Okay. So this talk is divided into 3 sections. Firstly, we'll be going over the background of this model called the unitary coherent particle filter. Secondly, we'll talk about a few of the results of the research, and then we'll go over, and then finally I'll go over what we've done and where we can go from here. So the unitary coherent particle filter was first developed in 2010 by Charles Fox and Tony Prescott. It was developed as a fully Bayesian implementation of a subiculum septum circuit within the hippocampus. We do this by using external stimuli and recurrent connections in the entorhinal cortex and distribute this information to lamella in a representation of the dentate gyrus. These are then sent into a representation of the CA3 subfield, which is modeled as a temporal restricted Boltzmann machine, before being decoded in the CA1 subfield back into our set of features, which we can use for localization or state estimation. Initially, these weights were set by hand in the first paper. However, as time has gone on and the model has evolved, we have used automatic assignment as well as a learned set of weights for starting off. So the first of the major equations in the paper is about the Boltzmann machines. This estimates a joint probability distribution from the observed and hidden states, and these are fused to denoise any sort of noisy inputs from sensors, which we can then use for localization based on the memory of the filter. So as part of our Boltzmann predictions, we use a modification to the Boltzmann machine, which computes a maximum a posteriori estimate. And this essentially collapses a Bayesian state into a true-false scenario, and makes it easier to decode. This then feeds into a loss of tracking detection, which is done by the subiculum and the septum, disabling the recurrent connections and prioritizing the current sensory inputs for relocalization. Finally, we have the wake-sleep algorithm, which is used for updating the weights of our Boltzmann machine. Within the two parts of the equation, the wake and sleep terms, we found that it requires an after depolarization effect as an after depolarization effect within the CA3, and we can use this as an explanation for a 10 Hertz theta oscillation within the hippocampus model. So I've talked about localization and whatnot. So in our current work, we are using the plus maze model as a toy environment for the localization and learning what the robot can see, what the agent would be able to see based on its position within the environment, storing that into the memory structures of the unitary coherent particle filter model, and finding location based on that. So so now I'll discuss some of the software engineering side behind the development, behind the work that we've done. So at the top here, we have the code for calculating Boltzmann probabilities as it was in the original version, where we used NumPy as a serial CPU-based library for maths. And at the bottom, we have transformed the code so that it does functionally the same thing as the top, but instead we are using TensorFlow, which is more suitable for programming on GPUs and using other tensor-based hardware accelerators. So how can we prove that these modifications are correct? That's where test-driven development comes into play, where we can essentially write a set of tests around the function to estimate, to just check its behavior and understand whether there's any side effects that are happening that we need to be aware of or fix. Then once we have got our new implementation, we can then feed it back into the test system, and if all checks out, then all of the behavior and all of the expected responses will be correct. As an example, we have here a 2x2 weight matrix and a 1x2 input vector, and this test will check that the Boltzmann probability function I showed before will spit out the correct outputs of 0.54 and 0.59 to 2 decimal places. So now that we've done all of our refactoring, let's test it. The initial run was when we looked at it on the CPU versus GPU, we saw a very large disparity between whether it was on the CPU or on the GPU. And digging into the code and what the processor was doing at any given time, we found that there was a lot of internal TensorFlow memory management stuff, which essentially led us to believe that a lot of this time disparity is to do with the transfer of data from system memory, which is relatively fast, to GPU memory, which is slower. So after seeing that and knowing that GPUs tend to work better when we have a lot of data that needs to be processed in the exact same way, we added in extra neurons in multiples of 1,000 to the CA3 and measured the time it took them to complete. There's very little difference at the 1,000 to 2,000 scale where each where CPU took around 20 minutes to complete, whereas GPU took 5. But when we get up to 20,000 neurons, there's a much different story, where the CPU is taking 850 minutes compared to the GPU only taking around 50 minutes to train and infer. No, there's a button. So now that we've got this, what can we do with it? Well, TensorFlow has the ability to scale across multiple devices, whether that's more GPUs on the same machine or actual computers. This should allow us to reach an even greater number of neurons within the model. Secondly, we can re-examine that plus maze environment and create some more realistic or complex environments for the filter to learn. And finally, we can take it from the simulation, as that is essentially what it is right now, to implementing it on a real world robot, whether this is for autonomous driving or other navigation, we are not sure yet. So in summary, the unitary coherent particle filter is a model of the hippocampus which maps a biological realistic neuron into Bayesian mathematics. The previous versions of the model work for robotic navigation in a toy environment, but it's not suitable on a real world scale. And as such, our work has made it suitable for a large scale environments via GPU acceleration. Thank you for listening. I'm happy to take any questions.
Thank you, Jack. Very nice. Great talk. So I have one question. I think we might be able to have one or two questions, depends on the time. So my question, and it's actually very relevant to the summary, is the following. So this previous model was made for robotic navigation. So how would you then scale or adjust this model via GPU that you have done for similar tasks where you can actually, let's say, compare between the previous and this current model?
So if I'm understanding your question correctly, you're asking how will we take this software-based model and put it in and either deploy it on an actual robot in a complex environment.
Sure, I mean, you can start from from simpler environments and go up, but how would you implement this on a real robot? Yeah.
So if we were to implement it onto a real robot, this is the personal approach that I would make, and I'd have to discuss with the other authors about it. But I would use I'd convert it to a ROS module rather than being a pure Python, and then we would be able to experiment with it in simulations, probably using a Turtlebot based robot initially before moving on to real hardware or cars, for example, cars. I hope that answers your question.
Yeah. I mean, would you expect also some improvements compared to the previous model, like better?
There may be improvements compared to the previous model. All of the environment loading is done right at the start beforehand, and it is a little bit on the messy side, so that may have some impact on the overall process. If that was to be cleaned up a bit or refactored, I'm fairly certain we would be able to see some form of speed up. Yes.
Great, thank you. Paul?
So thank you for your talk. But why would you claim that CA3 is a Boltzmann machine, for instance, right? So the fundamental question is, why does this model that you present qualify to be called hippocampal, right? So for me, if you claim that the model reflects the hippocampus, that means you make very specific anatomical and physiological commitments. And I don't see that's clear in this case. So for now, I would say your model might be an interesting model, but it's not a model of the hippocampus unless you show me what these testable commitments are. So could you comment on that?
Yes, I can comment on that. However, my role within this project and paper was more on the software engineering side of things, of taking it from being serial to parallel, and that sort of thing would be better directed at Charles Fox.
Mm-hmm. No, Charles, and I have these discussions also with Charles and with Tony, but then bring it back home, because you cannot just put anatomical labels on arbitrary machine learning algorithms, right? This makes no sense. So because, that of course also then has implications for how we can build on these models. Like, I can well imagine this is super interesting to have this speed up that you now have worked towards, which is great. But then, of course, what we would like to see is not so much linking to the biology, but let's link it to high performance alternative simulation environments to show how you are better or worse than those solutions, right? Like also in the HVP environment, people work really hard on different software simulation platforms, right? Like Nest, for instance, and Neuron and Brian and what have you. And to really start to benchmark against those, I think would then tell us a lot more than getting bogged down on whether this is really a model of hippocampus or not. So have you looked at this kind of benchmarking?
No, we haven't looked at that kind of benchmarking, but it is something that we will take into consideration in the future.
Well, I think it's essential if you want to move forward, right? Because these are your competitors, so we have to understand that, right?
Yes, that is correct.
All right. So, well, great project. So good luck. Thank you.
Thank you.
And one last question, William.
Hi. Interesting talk. This is just a very, very quick question. So you went from CPU-based workload using NumPy for just serial computation to GPU computation using TensorFlow.
Yep.
Assuming just because it's fairly easy to convert from NumPy-based to TensorFlow-based computation.
Yep.
My question for you is, the other kind of popular framework for this would be Torch or PyTorch-based GPU computation. Did you look at implementing it in that sort of framework, or do any comparisons, or just, I guess, why did you pick TensorFlow versus other GPU-based frameworks?
So my rudimentary understanding of PyTorch is that was more aimed at the artificial neural network. It's more designed for that sort of thing, whereas TensorFlow at its core is is more focused on the mathematical operations. That is one of the reasons we chose TensorFlow. However, as the purpose was to do the GPU scaling with test-driven development, time constraints led us to not being able to have an option of using PyTorch within the work.
Okay, that's fair. As just a suggestion for future work, PyTorch and just Torch does fundamentally also do just the mathematical kind of vector processing.
Yep.
It really just ends up being a difference in how kind of the graph structure is manipulated. So it could be an interesting benchmarking comparison for future work.
Indeed. Thank you very much.
Yeah, thank you for your time.
Thank you very much, Jack, for your talk. And now we can move to the final top paper of this session, which is Homeostatic and Allostatic Principles for Behavioral Regulation in Desert Reptiles, a Robotic Evaluation by Tu and Go. It will be presented by Tu and Go. William, if you don't mind removing the hand just to have it for the next one. And thank you, Tu.
So just let me know if you can still hear me and you can see my slide.
Yes.
With it.
Thank you. Yes.
Okay, perfect. So hello, everyone. So homeostatic and allostasis are known in various studies as unexplanatory fundamentals behind physiological and behavioral regulation. So in this study, we follow those principles to model the control system of a simulated robot that captures thermoregulation and foraging of reptiles as navigation profiles. So the survival of animals in general is accomplished through behaviors being regulated, not only respond to the events in their ecological context, but also to stabilize their internal states and then reduce their internal drives. And those drives often arise due to an unbalanced state of their internal environments, including many physiological conditions that are essential for viability, for example, glucose level or body temperatures. The term homeostasis, as coined by Cannon, refers to any autonomic or somatic activity used to preserve the constancy of those conditions. For example, in the case of temperature, the animals can sweat or avoid the heat to preserve their desired body temperature. And our study was inspired by the example of these Namic desert lizards. In general, reptiles lack a metabolic heat production mechanism as the endorphins. So they perform thermoregulation exclusively through interaction with their environments. So in the case of this lizard, once the surface temperature in the desert increase, The lizards can lift their feet off the sand to prevent the limbs from burning, but when it gets extremely hot, like 40 or 50 degrees, the lizard will dive into the sand dune where the temperatures are cooler. So in terms of homeostasis, this lizard behave to sustain a desired body temperature of 30 degrees. So homeostatic imbalance is known in previous studies as the primary assessment that allow organisms to respond to their internal rise. Computationally, physiological condition, for example, a glucose level, that can be represented as a homeostatic system, which defines an ideal range for that condition and evaluates whether the actual condition is within that range or not. And the state of an internal homeostatic system is classified as balanced when the actual values stay within that range. However, when the actual value goes outside of that range, the animals will be motivated to bring back balance. And that tendency, the intensity of that tendency to bring back balance, we call them homeostatic errors, is calculated by the distance from the actual value outside of the desired value range to the closest homeostatic bound. The next term is allostasis, which is coined by Sterling and Ayer. Is known as a solution to maintain a dynamic equilibrium of multiple drives being satiated and by orchestrating multiple homeostatic loops. However, the term is understood and applied differently in various researchers, so the mechanism remains unclear. We argue that allostasis does not necessarily imply choosing which behavior to fulfill foremost among many behaviors, or anticipating future needs through learning and then prepare to fulfill the needs before they arise. Instead, we believe that it can be retained as a reactive mechanism, coping with immediate fluctuations. And so on that definitions, homeostasis is a collection of first-order negative feedback loops involving corrective reflex. And if homeostasis fails to stabilize certain physiological variables within their viable boundaries, then allostasis will intervene. And many previous work, for example, the most notable work from Sanchez-Fliebler since 2010, focus mainly on adjusting the desired value or the set points of the homeostatic feedback loop. In the study, we aim to examine the interaction between multiple loops instead by changing their ways. The main reason is that an absolute reconfiguration from a desired value to another one leaves us in doubt, especially when the behavior change is coping with proximate stimuli in a constantly changing environment. So there are two mechanisms that we choose to dynamically weight the rise. The first one is allostatic load, which we chose to be the interoceptive way of modulating ways. Allostatic load is the accumulative burden of chronic exposure to unresolved homeostatic errors. And it is calculated as the mean value of all homeostatic errors during a specific time window. When I mean a prolonged period here, it means to compare to a normal condition when the animals can perform its homeostatic recovery. And we also want to compare that with external cue for adaptation, which is straightforwardly the number of resources that the animal can detect in its environment. So response modulated by this dynamic weighting mechanism are initiated by assessors and sustained for an appropriate time interval and then shut off when the thread is terminated. We took an idea from previous work that the weights or the priority levels of independent drives will likely form a pyramid similar to Maslow's hierarchy of needs, where some drives can take precedence over others. So in this paper, they propose a renovate hierarchy, and we choose it to incorporate in our study, focusing on animal needs modulated by homeostatic nature. One small note that all the needs within each layer might have different values instead of having the same value for all of them. And as we dynamically weighing rice, these layers should be overlapped. And the relative position of rice within the hierarchy modified in response to either exteroceptive sensing of external resources or interoceptive allostatic load. And with that, we believe that this regulation mechanism can offer better evaluation of the internal states and based on how much they match with the external threats or opportunities. So we have two hypotheses here. The first one is the dynamic reorganization of hierarchically ordered rice can allow the agent to achieve higher internal stability, which is the actual values, and also reduce the rise intensity or the homeostatic errors, than the fixed order of constantly weight rise. And then because we modulate weights based on two mechanisms, so we have the second hypothesis, which is an agent weighing its internal rise based on allostatic load can regulate its behavior as sufficiently as the one waving its drive based on exteroceptive sensing. And in order to analyze that, we analyze the behavioral trajectory and also the internal stability of the agents. So we started building our control system by decomposing it into smaller modules representing fundamental functions of action selection and behavioral regulations. This architecture is influenced by the distributed adaptive control framework with 4 layers stacking on top of each other, representing different levels of cognition and behavioral regulations. And our work will focus mainly on the reactive and the adaptive layers of DAC. So our cognitive modules will proceed exteroceptive information inputs from the environment, as well as generative allostatic load or rise intensity as interoceptive signals. And those information are integrated and used to weight the drive. So these allostatic controller modules will have access to an internal hierarchy of drives and then can modify it and make change to it and retrieve information for it to weight the rise. So all the drives are weighted and sent simultaneously to the action selection modules, from which will serve as a bottleneck, transforming those information into representations of behaviors and select which action to trigger. So there are 3 homeostatic systems implemented in our study, which is hunger, thirst, and thermoregulations. We construct the temperature gradient as a heat map, and the agent will try to place itself at an optimal distance far away from the highest peak. So the agents have local view comprised of 4 quadrants here, and the actual value is the mean of the whole 4 quadrants, the mean temperature values. In terms of navigations, we follow the paper from Sanchez Rivera to use XI and AD to control the navigation of the agent. I will not go too deep into this because of time, so we can also go back whenever you have questions. And for the energy and water homeostatic system, we build it without gradients and simply put water and food resources within the explorable space of the robot. And whenever the robot can approach resources, there will be some change, which is the impact aid here, that increase the actual values of the system. For the allostatic control, this is 2 ways of modulating the weighting factors. first, based on the number of resources detected. And in short, less resources, the weight will be heavier. And then based on allostatic load, in short, like if the animals suffer from higher stress, then the associated system will be weight heavier. And also at the end, all the weighting factor will be integrated with the right intensities, and the result, which we call like the weighted drive, will be the key criteria to determine which is the dominant drive, and then based on that, select the action to trigger. So first, as I explained before, we decide to analyze the trajectories of agents grouped by 3 different weighting mechanisms. And we try to evaluate their abilities to reach the necessary resources. We expect to see the robot make more excursion towards food and water resources, as well as avoiding the high temperatures. So to visualize and quantify the excursion, we build occupancy map for each group of agents. So the green mark here indicates the locations of food, and water is located here with the blue mark. So X cells here will contain normalized number from 0 to 1 of that area, that particular area being visited. So we can observe that both adaptive agents, the first one and the second one, were able to prioritize navigation towards both food and water. And at the same time, they tend to visit the cooler sides more often, while this agent with a fixed weight failed to avoid the high peak of temperature. We continue to build some Venn diagrams of time spent at specific location. The units of the numbers here indicates the percentage of time spent the agent spent at the certain location with food only, with food and water, and so on. So we also plot some plots of time steps spent at specific location in order to know which agent can make the most of their lifetime, avoiding high temperature or searching for resources. So in terms of occupancy of the place with low temperature, we can see that the interoceptive agent modulated by stress are the only group that can achieve more than 50% of their time step staying at the cool locations. And in addition, both these 2 agents were able to spend more of their lifetime satisfying at least one of their lifetime, at least one of their needs. So this agent spent 78.3%, and this one's 80.4% of their lifetime to obtain resource or avoid high temperatures. However, we could not achieve any results regarding whether the agent with dynamic weighting mechanism can really balance the trade-offs between conflicting needs, because we can see here the interoceptive agent can achieve a percentage of 42.9 of their lifetime staying at the optimal location, where there are at least 2 resources occupied. And this number is 40.9 for the exteroceptive agent, but 42 for the agent with fixed weight. To continue, we want to analyze the internal stability of the agent. So to see if the agents can really stabilize all the physiological conditions and reduce all homeostatic errors. Here we extract a series of time series for 3 homeostasis systems for each group of agents. The black dashed line here indicates the desired values of the corresponding homeostatic system. The gray dashed line is just to indicate 0.5 actual value. We have the dash lighter color lines as the come from individual simulations, and the mean of them is the color thick line. So the actual values are expected to stay above or around the desired value, and the animal can be considered in danger when it reaches the bottom of the graph, for example here. So we observe that the first two agents with dynamical weighting system can maintain the actual value of mostly around the DVs with very few outliers or drastic shifts that could be caused by the lack of resources or insufficient responses. Also, if we consider this temperature time series as a collapse of the trajectories into dimension, we can see that they tend to react more quickly to go back to the desired value. So for the two analysis, we can prove one of our hypotheses that the dynamical weighting mechanism actually can have the agent to maintain a higher internal stability. This is a box plot of all of the homeostatic errors grouped by the agent type. And the green numbers here are the mean width, and the black numbers are the standard deviations. Those results continue to support our hypothesis that the interoceptive agent and the exteroceptive agent can produce higher internal stability and reduce and minimize the homeostatic errors better than the one with fixed weight. Another note is that we run several comparative tests, such as Wilkerson ransom and Levin's test, and we noticed that all of them get very low p-values, probably because the huge amount of data we have from almost 21 simulation with 500,000 time step. We try to explore a bit more by calculating the efficiency and equality score of a group. So the efficiency score of an agent during a simulation is the average values of all the mean AVs from all homeostatic systems. And the equality score is calculated by subtracting the discrepancy between the AVs of the system with the highest and lowest AVs from 1. So here we can see that the interoceptive agent exhibit a slight trend of achieving higher mean score in both efficiency and equality compared to the other two agents. And we expect this to give an insight for maybe for future work into how this agent can actually better distribute the regulation efforts. And so I plot here two plots of how of a single simulation from X type of agent to see how the weight can be modulated during a simulation. And also besides that, we continue with a correlation analysis to quantify the relationship between several internal variables of hungers and thirst homeostatic system. So data was combined from both systems of each group and plotted into a single matrix. And X cells here contain the correlation coefficients of the two internal variables. So we can see that the interoceptive agent exhibit a trend to have all variables negatively correlated with the number of resources detected, especially the weight drive and the weight. On the other hand, the exteroceptive approach demonstrates correlation that somehow point out the irrelevance of the K factor in the reduction of rise. For example, there is a significant negative correlation between ride intensity and weight, which kind of doesn't make sense because I feel stressed, but then I will not weight it heavier. And and also there is a positive correlation between the number of resources detected and the weight ride, which is also the will to take that actions. So which proving insufficient adaptation to resource availability. So we think that this does not mean storing the quantity of rich target is impractical in the second case, but future works to consider the couples of state and action and reward instead of simply inverting the detect number of resource as the way we did. So we don't have a significant R number of 0.3 to establish any conclusion about how the interoceptive agent can adapt to fluctuating stimuli. But we think that longer simulations or better techniques to compute the allostatic load could validate our hypothesis about how the allostatic load might play a key role in an intrinsic adaptive system. So in summary, we have addressed in which way behaviors can be regulated based on complementary homeostatic and allostatic principles. We introduced a concept of drives organized hierarchically, where the priorities are modulated by either allostatic loads or external sensing. This dynamic weighting mechanism is proven to offer final matchings between the internal states of the agent and the external threats or opportunities in compared to a fixed order of constantly weight drive. Regarding the future work, first, there is no predictive regulation carried out within our study. So although we use previous fluctuations to follow a logic, the agents were unaware of upcoming events or upcoming homeostatic errors, and that therefore lack a way to deal with the uncertainty of environment. We think the future work could integrate a model into the full DAC architecture, where we have the higher layer of associative learning or long-term, short-term system memory. And then we can learn from utilizing information from the past and make rewarding decisions for the future. Secondly, our model solve drive conflicts by ignoring less prioritized drives, following the winner takes all mechanism, which results in the lack of a multi-purpose behavior, such as ensuring an optimal path toward the location of resource that at the same time avoiding high temperatures. So a possible solution could be to build multiple gradients that can integrate it. And they can be internally defined, and they also can be learned. They can influence each other, and then form a single observable behaviors. We think by doing that, we can underpin a more integrative arbitration process that will prove a more higher level of behavioral regulations. And that's it. Thank you. Please let me know if you have questions.
Thank you very much. Due to time constraints, we have room for one question, I would assume. Alejandro, would you like to ask?
Okay, this thank you for a very interesting talk. Just a quick question. So this I saw like this only includes like some like consumatory drives like hunger and thirst. Will this framework also be able to include like some non-consumatory drives like I don't know, aggression or like eventually like some sort of mating or something like that?
Yeah, so the idea is that we start with this study first with only one layer of the hierarchy. And why I introduced the hierarchy here is to show you the big pictures of many needs that actually can follow the homeostatic mechanism, including mating and everything. Actually, we also discussed in our lab that there are more needs that is mentioned in the original Maslow hierarchy about self-creativity or more epidemic needs. And there will be also a need to review this hierarchy of needs and to analyze whether they will follow the same mechanism or not, which I think in many cases of the higher needs, they will not follow the homeostatic mechanism because they will grow the more you learn, the more you want to learn. You don't want to stop learning, for example. Yeah.
Thanks.
Great. Thank you very much for this interesting talk. It was very good. And thank you all previous presentations. It was a great session. So we cannot have more questions. I think we can go for a small break. Alex, 10 minutes, 15, how much?
Yeah, let's do a 15-minute break.
Perfect. So see you all in 15 minutes. Bye.
Okay, welcome back. All the talks have been wonderful so far. This portion is the short talks session B portion, where we've had 10 contributors, 10 authors pre-record 3-minute talks of their papers. And then we'll have, so I'll play those, and then we'll have a Q&A for any of the authors in attendance, and we'll proceed from there. So let me get this started.
Hello, my name is William Zahalski from West Virginia University, and together with my collaborators Sasha Zill and my PI Nick Szynski, I wrote a paper titled Load Feedback from a Dynamically Scaled Robotic Model of a Chorosis morosus Middle Leg. Load
Sorry. Is that the first one? There we go.
Welcome to my talk. My name is Peter Kappel, and I will present today my publication about an application-oriented comparison of two 3D printing processes for the manufacture of soft gripper systems. While the flexible actuators for such grippers were in the past often produced by a labor-intensive casting process, the advent of flexible 3D printing materials makes now actuator designs possible that were previously difficult or even impossible to produce. The aim of this study was to compare two well-established but quite different state-of-the-art 3D printing processes you see right now here. Both processes deposit the material layer by layer on the platform, but differ in the used principle. Material jetting on the left is based on the deposition of droplets of photopolymers, which are cured by UV light, which makes it possible to print multi-material objects and also so-called digital materials with various shore hardnesses, and support material, which is helpful for large and complex overhangs, but also obligatory. Whereas FDM on the right is the printing method based on material extrusion of a thermopolymeric filament through a nozzle. In this study, we used TPU, a flexible filament, and no support material. Since such studies were, to our knowledge, made just for rigid objects, the approach of this study was to compare both printing principles for their suitability to manufacture flexible pneumatic bending actuators in an application-oriented comparison. Therefore, we developed a test bending actuator that can be printed in its basic design with both printers. So you see here right now. The FDM method limits actuator design by the lack of support material and its nozzle size, while PolyJet prints must allow the support material to be removed afterwards. We solved this by a wide opening for the basic design, and for the PolyJet printed actuator, a second closable opening for cleaning of the support material, and for the FDM printed actuator with an additional removable rigid support inlay that was printed separately. We characterize the actuators by bending angle and torque calculated from the tip force. You see here in the small pictures how the parameters were measured, and in the diagrams, the data from the PolyJet printed actuators in red and from the FDM printed in black. The results reveal distinct differences in their response to applied air pressure, especially the bending angle differs in regard to the used printing process clearly. The PolyJet actuators are more sensitive to applied air pressure, while the FDM printed ones are more robust at all, since they withstand higher air pressures before they fail. Here are my acknowledgements, also for considering me for this short talk, and thank you very much for your attention.
Hello, I'm Kodai Kotono from Kyoto Institute of Technology. I'd like to talk about evaluation of gauge generation in quadrupedal leg locomotion with changing anterior and posterior extreme positions. It is well known that disabled cats can generate various gaits. For example, a cat can generate and transit from walk to trot and to gallop gait by changing belt speed. Even when a belt speed keeps constant, a cat transits its gait as stronger stimulation to MLR. On the other hand, It is also well known that the limb controller is located in the spinal cord, and the spinal cord can change stepping frequency and gait. Therefore, we believe that the spinal cord has an important role in gait generation. We propose a constructive spinal cat model for understanding gait generation. Then, we conducted simulation with the proposed model and analysis of gait stability. Let me explain our proposed model. The spinal cat model integrates Polygon spinal transition model into the leg controller. Left is a schematic model of the spinal cord. Right is the controller as a central pattern generator. The leg controller uses leg load and hip extension and flexion as a sensory input for the stance to swing phase transition. Each leg phase is determined by 3 components. The first is the phase information of each leg controller. The second is sensory input to the leg controller. The third is inhibitory interleave coordination. Eventually, they lead to gait generation. Let me consider the sensory input for leg phase transition. The center of mass movement affects leg load. AP and PP distance affects hip extension and flexion. We evaluate what kind of gait was generated by changing AP and PP distance through simulations. Finally, we show gait stability analysis briefly. This column map shows what kind of gait was generated at each AP and PP distance. In the low AP distance, the model generates pace gaits. In the middle AP and low PP distance, the model generates walk gaits. In the middle AP and PP distance, the model generates trot gaits. Therefore, changing AP and PP distance affects various gait generations. To analyze the gait stability, we create the return maps and approximate surface. Then, we calculate the potential function at each PP distance following the previous study. As a result, walk gait is stable at low PP distance. On the other hand, trot gait is stable at middle PP distance. Therefore, changing PP distance alters the gait stability. I would be happy if you have interest in my research and come to Q&A. Thank you.
Hi everyone, in this talk I'll present our work, James Luck, Deep Reinforcement Learning Toward Bioinspired Control based on the Aplysia californica feeding. My name is Wenhan San. I'm presenting today on behalf of myself and our co-authors at Carnegie Mellon University and Case Western Reserve University. In this project, our goal is to train a deep neural network that can output motor signals in a simulated biomechanical model to recreate a comparable swallowing behavior as shown in our original neuromechanical model. For the simulation environment, we use the OpenAI GEM libraries to recreate a biomechanical model of Aplysia californica in 1D to control and recreate its feeding behavior. Specifically, we include 5 motor neurons and 5 muscle groups in this design. We use 5 Boolean inputs to represent motor signals for protraction, retraction, grasp closure, and pinch motion control at a frequency of 20 Hz. We calculate the corresponding muscle activities as well as the grasper and head motions and force on object. For the policy network, we use deep neural network to approximate the optimal action value functions or Q function using deep reinforcement learning, and the learn agent policy is just a maximizer of the Q function at a specific state. We train the agent for 500,000 training steps, and at a frequency of 20 Hz, that corresponds to roughly 7 hours of simulated animal data. To get the benchmark, we run the expert model in the simulation environment for 10 episodes and record its performance. By training the DQN policy in the simulation environment for 500,000 steps, the learn agent achieved expert level performance, and we confirm that the cumulative reward of the learn policy gradually increase and converge to that of the expert level. By comparing the behavior between the original Boolean model, the expert model, and our DNN learn policy, we confirm that the DNN achieved performance that is comparable to the original model with qualitatively similar motor neuron control and muscle activities, as well as grasp motion and force on object. In addition, we demonstrate that the DNN shows robust behavior in previously unseen environments with variable strength seaweed. In this case, we increased the seaweed strength in different test cycles, and the learn policy was able to use higher forces and spend longer time swallowing the seaweed when the seaweed strength is higher. We would like to thank the support from our collaborators at Carnegie Mellon University and Case Western Reserve University, and the funding from the National Science Foundation NeuralNext program.
Hi, I am Alejandro Jimenez from the University of Sheffield. Today, I am going to present our work on cognitive architecture as a service, scaffolded integration of heterogeneous models through event streams. The use of socially assistive robotics have been shown to be beneficial for humans in different settings. For example, as companions, they can improve physical and psychological health by means of interaction or by inducing behavioral change. The success of those applications depends in many cases on the robot's degree of autonomy. In order to attain autonomous behavior and a successful human-robot interaction, we built hierarchical biomimetic architectures that are inspired by the mammalian brain. In this work, we propose to use motivation as a design principle to structure such architectures. Another technical contribution in this work is related to the communication schema between different models that compose the architecture. Those models are usually created by different labs using different technologies. Instead of communicating directly, we expose the whole cognitive architecture as a service that the models can consume. This concept is implemented using Apache Kafka, and the whole cognitive architecture acts as a service layer to the neurorobotics platform. Overall, it provides a pre-existing scaffolding that is motivation-centered. This means that there is a motivational controller that activates different motivational systems that are structured into search, pursue, and consume action patterns. As an example, we implemented a simple Breitenberg vehicle with two motivations. The robot decides whether to search or pursue the different targets depending on its motivational state. First, the robot's camera output is fed into the perception stream. The perception stream is processed by different perception models that contribute information to the where and what stream. This information is combined into the motivational streams using the motivational state. Finally, the action selection module selects between exploring and pursuing depending upon the detection of the stimulus. In the end, the robot's behavior becomes an oscillation that is coupled with the environment. Currently, we are continue testing this concept with different models and in a variety of use cases. And we think it has the potential and flexibility to support new advances in socially assistive robotics. Thank you.
Hello, I am Dr. Alex Hunt, and I am going to present the work of one of my PhD students, Cody Scharzenberger. Cody got married a week and a half ago. He's been a little busy with his honeymoon, so unfortunately, you'll have to hear the presentation from me. I'm going to talk about a functional subnetwork approach to multi-state central pattern generator phase difference control. So it's a very mathematical title. What do I mean by this? Well, we all know that many rhythmic movements are controlled with central pattern generators. This includes breathing, digestion, flying, paw shake, and locomotion. Diving into locomotion, it's often modeled with the classic half center. However, we know that there may be more than just two phases involved. We don't know exactly how many nodes there are in central pattern generators of spinal cords. And so we have developed some mathematical tools that enable us to make any size CPGs or any number of nodes. And for example, here you see a 4-node CPG where all the 4 nodes are connected in a way that's similar to the classic half-center approach, and we get alternating behavior from these nodes. Now, in addition to that, it's theorized that locomotor CPGs are connected directly in the nervous system, right? So, for example, if there is a central pattern generator for each leg, then there can be some connections between these pattern generators that help govern things like gait. And so if you kind of look at the different gaits that an animal might have, you see different phase differences between the legs during different gaits. So a walk will have this type of phase difference. Trots will have a couple legs in phase with each other and out of phase with the other ones. up to bounds, where you have a whole new set of phases. So we've expanded our tools to enable us to program the phase timing between the CPGs. So here you see we've got two separate CPGs, and we can control the phase between these with a central kind of command neuron. So this command neuron tells us whether we want one CPG to lag the other, and we can see that when we lower the voltage of this neuron, that the CPGs begin to become out of phase with each other. And then we raise the voltage of that neuron, and then they swap phases. That is kind of the overall gist of it. There's a lot of math and stuff behind this that you can find in the paper. Current process is also a little bit complicated, so we use the functional subnetwork approach and deliberately program in things like subtraction, integration of signals, modulation, and sum this all together in order to create the phase difference that we want. And so future iterations will look at simplifying this.
Hello everyone, my name is Riddhi and I'm going to present my research, a scalable soft robotic cellbot. Taking inspiration from cells on how different cells can combine to perform a task. Can we combine different robot units to modify its shape to travel efficiently over different frictional surfaces? For this preliminary work, we combined multiple soft units in a row to explore their behavior over surfaces of different frictional coefficients. Our model is inspired from Golostonian swimmer to move one cell at a time. We see the interaction of these units with the ground, which results in a nonlinear behavior. The F1 and F2 represent the actuator forces. We extended our robot model to combine different number of cells in the row. We saw very interesting behaviors over different regions of friction. For low friction regions, there was actually a slipping in the robot, and hence, with the increase in number of cells, the net displacement reduced. Whereas with increase in friction, the net displacement was higher as more number of cells provided greater stability to the robot. For a robot model, we combined balls with linear actuators between them and saw their movement on low friction and high friction area and for different robot parameters. For our physical robot results, with an increase in friction, the displacement reduces. With an increase in number of cells, the displacement increases, which is consistent with our results of the A4 region of the simulation. The robot model presented here is robust and has a simple control, which makes it a low cost and low complexity robot model. This research is useful to understand how many units can be connected or how the robot material can be designed to optimize robot locomotion on different frictional surfaces. Thank you.
Hello, I'm Kotaro Yasui from Tohoku University. Today, I'd like to talk about simple reactive head motion control that enhances adaptability to rough terrain in centipede walking. Centipedes have flexible bodies and many legs. Utilizing such body characteristics, they can walk effectively even on unstructured environment. So our motivation is to capture the essential control mechanisms that enable their adaptive walking motion and apply it to the controller design of multi-legged robot that can navigate complex terrains. As shown here, their central nervous system looks like a decentralized ladder-like structure. where segmental ganglion controls the motion of each body segment. So in our previous study, we proposed a decentralized controller for each leg motion based on the results of behavioral experiments. The proposed model consists of 4 control rules that determine the leg motions. First, when the leg touches the ground, it swings backward and kicks the ground. Second, when the leg loses ground contact, it lifts off the ground and swings forward. Third, when the leg has swung forward enough and its anterior leg touches the ground, it moves to the ground contact point of its anterior leg. Finally, when the abdomen touches the ground, the leg extends to contact the ground. Using this decentralized leg control, we succeeded in reproducing the adaptive centipede walking on various types of uneven terrains. However, this model still has limitations. That is, the simulated centipede sometimes became stuck when walking on irregular terrain with many gaps. This is mainly because the head collided with an obstacle in front. To tackle this problem, in this study we proposed a simple reactive head motion control. Specifically, we implemented a control rule that a few anterior body segments actively bend. As shown here, when the head is pushed against an obstacle in front, the anterior body segments are raised by active bending. Then, when the head collision is resolved, the anterior body segments start to bend in the opposite direction until the target angles for a straight posture. Finally, we verified that the newly proposed reactive head motion enabled the simulated centipede to negotiate with obstacles and traverse a very severe terrain with less scaffolds. So we expect this simple head motion control enhances the terrain adaptability in centipede walking. In the future, we'd like to test this control scheme in the real-world environment using centipede robot. Thank you.
My name is Clayton Jackson, and I'm a PhD candidate at Case Western Reserve University in the Department of Mechanical and Aerospace Engineering. I work in the biologically inspired robotics lab, and today I'm presenting my work entitled Canonical Motor Microcircuit for Control of a Rat Hind Limb. So the neural circuit used for locomotion in mammals continues to be a topic of research, but a prevailing theory is that central pattern generators, or CPGs, drive the motor neurons to produce the repetitive and rhythmic motion. One way we can model a CPG is shown here on the left with these 4 neurons. And we can see that if we model the half-center outputs, we get this nice repetitive motion. In addition to the CPGs, there's a variety of lower-level circuits that also play a role in locomotion. One of these lower-level circuits was first introduced by Holtburn et al. In 1979 and has shown up in a variety of other works. So specifically here, we're referring to this circuit highlighted on the right. So we refer to this circuit as the canonical motor microcircuit, or CMM. And this work focused on tuning a CMM to control the hip joint of a rat model to match animal data of rats walking on a treadmill. To do this, we first modeled the CMM in a software tool called AnimatLab. The neural models here use a non-spiking leaky integrator model, and the equations for that are shown on the right. Additionally, we created a biomechanical model for the CMM to control. So for this, we used simple flexor-extensor muscle pairs using linear hill muscle models. So the equation for tension there is shown on the right, along with a little bit more detail on the muscles. And then it should be noted that the model is air stepping. So the pelvis, which is shown in red, is fixed in place, and the leg does not come into contact with the ground. We then tuned the neural model so that the hip joint trajectory matched that of the animal data, and as you can see on the right, we were successful in replicating that oscillatory motion of the hip joint with the CMM. So some areas where we see some differences could be caused by a couple different things. One, simplifications to the biomechanical and neural models. as well as we see there's a larger error in the stance phase than in the swing phase, which could be caused by the model air stepping rather than having external forces from the ground. So some future works include developing an automated parameter search so I don't have to hand tune the model, and continuing to add complexity to the model. This work is supported by National Science Foundation NeuroNext grant. Thank you for listening. I appreciate your interest, and I look forward to your questions.
Hello, my name is William Zahalski from West Virginia University, and together with my collaborators Sasha Zill and my PI Nick Szynski, I wrote a paper titled Load Feedback from a Dynamically Scaled Robotic Model of a Corosius morosus Middle Leg. Load sensing is critical for the control of walking in animals, including insects. They have evolved several sensory organs and neural systems to aid them in this behavior. One such class of organs, the campaniform sensilla, is the focus of this paper. Thanks for decades of extracellular recordings of campaniform sensilla neurons, we know that they respond to loads on the legs in a highly dynamic way, first responding strongly to the onset of force and adapting to a constant force over time. However, some recordings are difficult to collect due to many factors, such as interference from muscle potentials and the placements of campaniform sensilla on this insect being inaccessible in some cases. This is why, to better understand what load the insect experiences and what sensory feedback that the nervous system may see during walking, we've constructed this dynamically scaled robotic leg, shown in this figure. And on the robotic leg, we've affixed strain gauges in the same positions and orientations as the major campaniform sensilla groups, such as group 3, 4, 6A, and 6B. As you can see, we have some strain gauges attached here. We also have the leg on a linear guide, and we had a step on a treadmill. The linear guide is used to simulate the weight and movement in the insect body, while the treadmill allows the leg to simulate walking. Here you can see the linear guide on its rails. And here's another figure showing a little bit more detail about the location of the strain gauges. So once the strain gauge data is collected, it's run through a dynamic model of campaniform sensilla discard developed in a previous study. So our study reveals this loading patterns experienced by the leg even as the weight and joint stiffness is altered. As you can see in this data, this is just the baseline configuration. But as we add weight, sure, the strain changes in an order of magnitude, but the shape remains the same. And we also even were able to change the complete gain of the motor, and we still retain this shape. Additionally, our simulated campaniform sensilla strongly signal the beginning and end of stance phase, which are two key events in the coordination of walking. Here's some model outputs. Here's one in the baseline configuration, still showing these strong beginning and start of stance phases. Here's one with the 500 grams added, again, strongly showing these spikes. Here's one with 1,000 grams. Now there is a bit of a dip here from this ripple, and that can be explained in the Q&A. And here's again with the 2 XP gain. Now I really appreciate your time, and if you have any questions, please come to my Q&A or email me at wz0007@mix.wvu.edu.
Hi, my name is Chanel Picard, and I'm going to do a presentation on time evolution characterization of behavior class prototypes. So the motivation for this work actually comes from my last paper where I quantified the differences in behavior structures of hunting praying mantises that were observed in various feed states. That is, from the starved state up through 4 food items that were consumed, and such behaviors such as translation, rotation, etc. were able to be extracted from videographic data and generate these ethograms. These ethograms then lend themselves very nicely in generating transition probability matrices, where each behavior has an associated probability of transitioning into every other behavior, including itself. So for a given feed state that has many sequences, each observed sequence can generate a transition probability matrix, which then actually turned into an interesting question. If I have many transition probability matrices representing the zero-fed state, how do I find the average of that? And mind you, this transition probability matrix is a proxy for a time-dependent sequence, so how might I then find the average of that? Which again is generating a prototype, which is defining a central tendency. In order to go about answering this, I used 3 different techniques in generating a central tendency. This included the cumulative approach, which was literally just finding the mean of each transition probability across all the samples within a feed state. So then for each of the feed states, I generated a unique prototype using the cumulative approach. This was done for the centroid as well as for the madoid techniques. And it's interesting to point out that the madoid is actually unique here in that it actually generates a prototype that is an actual data point. That is not the case for these other 2 techniques that generate an abstract representation of central tendency. And as a side, I picked centroid and madoid techniques because they are commonly used in clustering techniques. Several analyses were done in the paper to give an idea of different time evolution metrics for these behavior sequences. However, I'm going to focus only on the hit times here, and that is the time that it takes to first reach a specific behavior state given a starting behavior. And looking at this graph here and being a little unorthodox and starting at the bottom, the madoid actually has these very egregious discontinuities, meaning that these behaviors that we see along the x-axis are actually never reached. So then looking at the centroid, we do reach all the behaviors, but we don't really see any clear delineation across the feed states that give insight into trends. But now looking at the cumulative, which is this top graph here, I'm actually seeing that there's a trend across satiety. So starting with the lower feed states are on the bottom, I start to see this gradual increase as satiety increases as to the amount of time that it takes to reach any given behavior. In conclusion, this paper covered 3 different ways of finding central tendency. These 3 ways were not equal in capturing time evolution trends of behavior sequences. And in this paper, as well as a little bit here, it was covered that cumulative prototypes best captured empirical trends.
Okay, excellent. Thank you all for your presentations. We can now open up the floor for questions for our presenters. Yes, Vicky, I see you have a question.
Yeah, thank you. Very interesting talks. I have a question for number 4, which is cognitive architecture as a service. It's a very interesting approach, especially the service part. And I would like to know what are the limitations of such an approach, as well as how would they be implemented, for example, as the others kind of mentioned, into a social context with social robots. Thank you.
Yeah, thank you for your question. Yeah, I think that this shifts the attention, like not from like before, like in many approaches for integration, you have to find the right format to communicate with different models. This kind of shifts the attention to find the right data format for the streams, or what kind of information and representations are good in a specific context to keep in the streams of data or streams of events that are happening. So for example, if you're going to represent an action, what information do you need for that? But the benefit of this is that you can include way more information into the stream, so you are not limited to the actuators or sensors of the robot, but you can actually include information from other devices in a house, like in a social environment, or even from other robots that can fit into the same streams, into the same cognitive architecture of the robot. So so I would say like one of the limitations is, yes, like the complexity is still there, like the complexity of communicating, for example, spiking models and firing rate models, still there. It just translated into finding the right format that's gonna be useful for the cognitive architecture as a whole, and the models have to satisfy. I think right now we are in the process of discovering those difficulties and trying to see how we solve it. Like, for example, we are trying to integrate some hippocampal models, and then we see, okay, what kind of representations are useful? How can we use the information that this model provides inside the architecture? And also we have like some visual system models, some depth perception and saliency, and all this information. The idea is that eventually it finds the way into this cognitive architecture as a service and can be consumed by other models. And in a social environment, it's also, I think it's like a process of discovery of I think that the approach overall is very scalable. So we test like different kind of use cases, and then we see like, okay, is this, what else do we need, or what is where do we find like a wall that we need to try to use to expand the concept.
Yeah, because I can imagine that if one of the important parts on this is, for example, the motivation and the drives that you have implemented there, if we want to, let's say, scale this up to social robots, then you need to implement slightly more complex, let's say, drives and motivators. And when it comes to the social approaches of robots and the different needs that are not necessarily going as simple as the ones that have been presented. So yeah, that would be very interesting to see how that approach would go.
Yeah, like we are combining this work with other models of motivation. I think in general, as with the previous talk on the pyramid of needs and stuff, is our approach is in general like find like a very fundamental try to mirror how the brain process like motivations, trying to model like a lateral hypothalamus and this approach and avoid pathways. And ideally, it will be to build the whole pyramid on top of this very primitive system, kind of in an hierarchical way. So that like more complex motivations, like let's say affect detection, or like some more like socially driven motivations can be can kind of take over this very primitive system and use it to build on top of that.
Great, thank you very much.
Okay, thank you, Vicky. Thank you, Alejandro. Oscar?
Oscar, we cannot hear you. I think you're unmuted, but we cannot hear you.
Okay, we'll come back to Oscar. Roger, did you have a question?
Yes, I have a question about the scalable soft robot, robotic cell bot. It's kind of cool. The ball is connected with linear actuators. I assume. Well, my first question is that do the balls move by sliding or rolling? It looks like they're sliding. And second, does it move by peristalsis? Does it move by peristalsis like a worm or a robot? Is that the point?
Yes, so right now the balls are connected with linear actuators between them. So each time we actuate them, they slide. Yeah, we have like metal bars on the side to just right now explore like the simple linear behavior and stop them from rolling. I think that was like the sliding one.
Okay, thanks.
And for the we were inspired by the Golostonian swimmer. It's mostly in like fluids where the balls there was like how like there is like movement. I showed like there are different balls, but here we see them on ground. So on the ground there is like this frictional interaction between the cells and the ground. So this the friction provides like a force to the balls, which leads in a nonlinear behavior of the balls, which gives these interesting behaviors over different regions on how they move. Do they, because sometimes they end up, because of the friction, they end up moving in, slipping back a little bit as well.
Okay, thanks.
Thank you. Okay, it seems Oscar, you fixed your mic. We'll go back to you.
Yeah, thank you. Yeah, I was following with the question of Vicky in the same interest. It's also about the motivational model of Alejandro. So my question is because I think this is also quite related to the word of the that we saw before of TUE and how we manage different drive. And the question was how hard doing this dynamic of the drive. There is a like a drive reduction when the behavior is done, or this is depending of another thing, and also if this is scalable to out of drive. Like for instance, now we have seen like two. If Alejandro think that this could be scalable to many others drive.
Yeah, yeah, like the dynamic control, like at least one version of it, like is we presented like a first version of it, like in the previous conference last year or like last 2 years, and we are finishing the paper. It should be out like soonish about. So our first approach for it is like having like a dynamical system, which is like a relaxator oscillator that switches between the two motivations based on the some internal dynamics of any need, like some buildup of some energy or like some physiological system, but then it also depends of like some input from the environment, and this input, like initially it just depends of the satisfaction of the drive, like reaching like as in the previous talk will be like just reaching the resources, and then that will just replenish whatever dynamical phenomena is happening the lower in a slower time scale. But as you say, in the robotics, and it's also in animals, like you can also satisfy the drive by just the action, like just performing the action, performing certain behavior. So it depends. It depends what kind of drives and the extending this to multiple motivations is in principle viable. Like we are also exploring that option, but it has also like some complexities, and I think that's also not clear from biology, or at least my perception of that is like was the right of having like multiple drives in this dynamical setting, like because some of the drives are organized like in some hierarchical manner as we saw before, or are they actually part of the same phase space, like attractors in the whole phase space? So yeah, I think that brings a lot of questions that needs to be answered, and I think we are in the process of trying to find.
Yeah, exactly. Yeah, I was wondering also that how do you select the drive? Because also for me, like, would make sense that the social robot should be endowed with a sort of theory of mind, so that the drive that he is attending is not their own drive, but the drive of the person that is in front. So you are trying to fulfill the necessity of the other person. I don't know if...
Yeah, like in that, like we have another model, like we are also studying this concept in a model of attachment, which I also write in the paper. And I think that's the whole, I tried to put it in the talk, but there was not enough time. I think in this concept of dynamical systems related with motivation, kind of behavior becomes some sort of oscillation. And then when you interact with the other human or the other, that becomes like some sort of coupling between the two oscillators. So in the attachment model, for example, you have like the robot has some need to receive care, and it estimates somehow the phase of the oscillation of the carer, of the parent, and then somehow you can synchronize these two oscillators so that they, whenever you perceive the other agent to as able to give care, then you kind of try to synchronize your natural oscillation. Because I think like in animals it's something like that. You have like a time budget for different actions, and you just oscillate between them. So that's more or less the kind of like the one of the consequences of this approach, at least.
Yeah. Well, thank you. Wonderful. I will looking forward to see that coming up work. So thank you.
Thank you for your question.
Okay. Thanks. Alex, you have a question?
Yeah, my question is for the 3D printing of pneumatic bending actuators. And so I had a couple of questions around it, actually. The first is, if you've got kind of the one system used much lower pressures, but you kind of talked about durability based on high pressures. And I was wondering if you looked at durability kind of just based on bending angle.
Okay, yeah, hello. Actually, we did not explicit looked on the bending angle, but it was like that the polyjet printed bending actuators failed before they reached a lot more. So we tested the actuators until around 1 bar of air pressure, and then it stopped because they always failed. And with the FDM printed actuators, we just tried out how far we can go. And this was always around 4 bar and sometimes a bit more. Yeah.
Okay. Just kind of like feeling them, are the FDMs just feel physically more durable?
Yeah, they were actually, I think the main difference in the material is that the FDM printed are more stiff at all. And so I think this is where the durability comes from, yes. Okay. While the polyjet printed were at all more softer, so they were more sensible in their response, but also they break earlier and had a lot of material failure, yeah.
Okay, and then another question is, we use some FDM printed parts with our McKibben type actuators, and we get air leaking through them. I don't know, question if you saw any of that happening.
Actually, this is more the work of my colleague, Stefan Konrad, who did a presentation yesterday. And we tried a lot of stuff out in the sets in the setting for the 3D printers. And first of all, yes, we had it, but we tried it out until it was good at all. And we could go to membrane or thicknesses around, I think, 0.6 millimeters, and usually they don't lag if the printer works good right now. Yeah. So there are some publications from him from last year and 2 years ago, I think. So maybe you could take a look into it.
Yeah, that sounds great. Thank you.
Yeah. Okay. Thank you for your question.
Thank you. Any other questions from the audience? I have a question for the simple reactive head motion control on the Centipede. I was just kind of wondering, as I listened to the talk, how much of that was biologically inspired, or was it just kind of like, okay, we know that we know that we're running into this problem. What can we do about it? Do you think that's kind of how the animal approaches the problem, or if you could talk a little bit about that? Thanks.
Yeah, thank you for the question. So our decentralized leg controller is based on the behavioral experiment. So we actually use a centipede to observe their leg motions. So this is very biologically inspired. So, but in this study, we proposed active head motion, but this rule is not based on the observation. So in the future, we'd like to see how they use the active head motion. Yeah, so but my intuition is that they are using a very nice flexible antenna at the head, and it might be similar control strategy with some kind of literature in the cockroach locomotion. Yeah.
Yeah, no, that's great. I was just kind of curious. I was wondering, so do you have in the works, do you have an experiment planned with the centipedes to try, to try to make some of these observations and see if you can, draw a comparison between your control strategy and what they do?
Yeah. So we'd like to first see the actual head motions, how they oscillate the anterior body segment, well, with antenna sensing in relatively simple unstructured environment. Yeah, that's the next step.
Gotcha. Yeah, cool. Very neat.
Yeah, thank you.
Okay, I'd say maybe a couple more questions if anybody has any. Otherwise, we can. Alejandro, yes.
I think I have one for number 10. Just like a quick one. I think it's a very interesting approach. Do you think this can be used to like generate behaviors? Like I don't know if you mentioned this in the talk. Maybe I got a bit distracted, but I just see like this has the potential of generating behaviors like for some robotic agent, like to just take in the prototype and just try to introduce some noise or variation, and then just kind of you can have like a very rich set of behaviors based on these prototypes. I don't know if you have any comment about that, like the potential uses of this.
Sorry, no, I had a little difficulty hearing the question. So was it adding noise to the?
Yeah,
sorry,
no,
I don't know why this speaker is on the floor, but I'm using someone's office.
No, I just find it like very interesting, the concept of having like a prototype for a behavior, because I think that's something that people have been looking also like a lot. So I just like, I don't know if you have any comments about what potential uses these kind of prototypes can have like for the future, like for robotic agents. Like,
yeah, no, yeah, that's a really good question. And primarily my research is in assessing the behaviors of animals and for various reasons, but one question that might arise is that if you're trying to build or construct the most used approach to solving a problem, and a problem may have a multi-step process to that. So again, a behavior sequence in this case was an animal that's going through a series of actions. So turning, rotating, translating, and doing that in various combinations for various time bouts for each of those things. So then the question might arise is that if you are wanting to capture an action, maybe let's say for training robotic actuation for a certain task, the best way of flipping an egg, and looking to the chefs, the great chefs of the world on how they do that, and you monitor and do a time series of how they go about doing that, you might want to then capture the most consistent way in which they're doing that. And, you know, in everyday verbiage, we call it an average or a central tendency. In this case, I'm calling it a prototype. And so in such cases, it's a little difficult because going about and solving that problem, again, being a time series thing, from iteration to iteration that you do this task, you might do different, you might have a slight variation in the amount of time that you do each type of task or each portion of that task. So you might spend a little more time in the elevation of the arm. The rotation aspect might take a little bit longer from time to time. So it becomes a little more difficult when doing like estimation of parameters if you use multivariate approaches on time series data. So in this paper, I didn't directly compare time series or like a sequence to a sequence or time series to time series in large part because the time series had a lot of variability. So there was not a lot of overlap. So I didn't use a lot of the standard approaches that maybe some of other people in the audience who are familiar with time series analyses might do, such as like vector autoregression or ARMA, where you use a moving average. So I summarized the data in this case, doing it with transition probability matrices. And that's ultimately what I was trying to do. So by generating this transition probability matrix and finding the quote unquote average transition probability matrix, you've now created more or less a template to create a average time series thing through maybe like simulation and stuff like that. So as far as utility, I can think of something like that, trying to train in the sense of robotics for specific tasks.
Yeah,
I don't know if that answers your question.
Yeah, no, yeah, it's just interesting because like one of the themes in motivation, which is my area, is just how to encode like action patterns in robots that are like very stereotypical things that animals do, but they still have to be different, like it's slightly different each time to accommodate. So yeah,
so even stereotype, no, you're absolutely right. Even stereotype sequences will have variation between them, right? But and yet we still call them stereotyped approaches to it. In this paper, I did hunting praying mantises, and yeah, we broke it down so that you can isolate and identify stereotyped regimes of behavior. However, that still is noisy. That is still variable. So I think you asked the question on noise, introducing noise. I didn't introduce any artificial noise into the data set. So no, I didn't do that. It's interesting. Yeah.
Thank you.
Yeah, thank you for the question.
Okay, great. Alex, how are we looking on time? Should we break for 15 minutes or so?
Yeah.
Okay. Okay, I guess we'll break and start up with the last set of talks in about 15 minutes.
Okay, hello everyone. I think should be starting soon. Or just give a couple of minutes. So I think we should be ready to start, right? Okay, so welcome to the third, no, the final session of today. We're gonna do like 3 very interesting talks, and we're gonna start with Clarissa Goldsmith, analyzing 3D limb kinematics of Drosophila melanogaster for robotic platform development. Welcome, Clarissa.
All right, thanks very much. Let me just get this shared or presented.
Sorry, we cannot.
Can you not hear me?
Oh, sorry, I forgot I have my. Okay, now the floor is yours.
We see your notes. You may
Yeah.
want.
There we go. Just gonna swap them, I think.
It's good.
Yeah, we'll go with this. All right, everybody. Hello, my name is Clarissa Goldsmith from West Virginia University, and I'll be presenting our recent work on analyzing 3D limb kinematics of Drosophila melanogaster for the development of a robotic platform. I'll begin with a little bit of introduction. There we go. So the work that we do in the Neuromechanical Intelligence Lab at WVU primarily focuses on using robots to investigate the nervous system. Over the last several decades, various degrees of biological inspiration have been used to improve robotic capability. And as Paul was kind of addressing in the keynote questionnaire today, in turn, there's been a recent development where highly biomimetic robots can help test biological hypotheses. In this endeavor, it's very important to have a high degree of biomimicry in the platform so that there is a minimal mismatch between the controller based on the nervous system and the structure, as it's not presently known the full intricacies of how the mechanics of an animal might interplay with the nervous system and vice versa. So that makes sure that the findings on the robotic platform are most broadly applicable to neuroscience as a whole. This comes with challenges, however, as robots have a lot of different criteria and challenges than animals. Robots are generally restricted to the technological state of the art, where animals don't really have that restriction. One common problem in this vein is that muscles are a lot more compact and lightweight than a lot of conventional electric actuators, meaning that if you're trying to implement all of the biological degrees of freedom in a robot, you might run into weight problems, making the robot, for example, too heavy to actually stand. So often we engineers, when confronted with this problem, will try and reduce the degrees of freedom to a minimum of maybe around 3, which is the amount you need to effectively move an end effector in 3D space. However, it can vary the biological accuracy of that simplification. Kind of along this vein, previously I worked on developing Drosophobot here and presented this at the 2019 Living Machines. This is a robot based on Drosophila melanogaster, the fruit fly. And as I kind of mentioned, one of the simplifications we made when building this robot was that we only included 3 leg degrees of freedom. And these ended up more resembling the stick insect than the fruit fly due to the limitations of even understanding the precise degrees of freedom of Drosophila at the time. But there have been a lot of recent technological developments to help us closer analyze the degrees of freedom in Drosophila. So the ultimate goal of this work was to identify the minimum degrees of freedom that would help best approximate animal movement in order to update our platform and continue our investigation of the nervous system. So with that said, I'll move into a little bit of how we did this. We used Deep Lab Cut along with the setup on the left here to get 3D pose estimation data for the fly. The fly was suspended on top of a spherical treadmill, suspended on a puff of air, similar to an air hockey table, so that there was minimal friction as the fly attempted to move along the ball. We used Galfor CS Crimson flies so that a red laser pointed at the head of the fly would initiate fast, sustained forward walking. And then additionally, we recorded their movement with 6 high-speed synchronized cameras that allowed us to get a whole kind of 3D view of the fly as it walked. So then we used Deep Lab Cut, as I mentioned, to get pose estimation data for 6 different areas of the leg. This kind of corresponded to a lot of where the joints were, or at least the union points between 2 different segments of the leg. So those were the thorax coxa joint, the coxa trochanter, trochanter femur, femur tibia, tibia tarsus, and then the tarsus tip. So using this data, we then generated a simulated leg chain for each frame of the video data. It's important to note here that we did end up leaving off the tarsus tip for our investigation, as the tarsus undergoes a lot of very complex movement during motion, and we were mostly interested in the motion of the joints higher up the chain, so it simplified our calculations to consider the tibia tarsus as the end of the leg chain. So we took these pose estimation data and then generated a leg chain shown here. This ended up having 7 total degrees of freedom. There were 3 at the thorax coxa, kind of representing a ball and socket joint there, 1 at the CTR, 2 at the trochanter femur, and then an additional degree of freedom at the femur tibia. We then used this 0 configuration leg chain to find a best fit configuration of this leg chain to the animal data for every frame. First, in a baseline case where all of these 7 degrees of freedom were mobile, and then we additionally started fixing degrees of freedom at the average value that they were exhibiting throughout a trial, and then observed the effects that that had over the course of a trial to the fit to the animal data. The way we generated this leg chain was using product of exponentials. This method involves calculating the position of any one of these points given a variety of angles from a 0 configuration of each of the degrees of freedom. Each have a matrix here. They all multiply together based on the different degrees of freedom, and their deflections multiplied by the 0 configuration of that point in space allows you to get the end point. Each of these exponentials here represents a twist matrix, which essentially categorizes the rotation of the degree of freedom frame as well as its movement in space from its origin position, using both the deflection of that degree of freedom, the rotational axis omega, and then V, the origin of the frame in the spatial reference frame, marked as O in this figure here, in the 0 configuration. So this allowed us to very swiftly get where the position of the leg would be for a set series of deflections for each of the degrees of freedom. We then embedded this function for the position of each point into an error function for each of the points, which is effectively just finding the distance between the point in the animal data and the point in the simulated data, and then normalized by the length of the proximal segment. So in the case of the CTR, this would be the coxa, as well as a weighting that helped us mainly just ensure that the construction was attempting to prioritize fit towards the end of the leg chain. It's also crucial to note that the segment lengths were recalculated for each frame of the animal data to attempt to minimize how much different frames of Deep Lab Cut, slightly altering points, might inflate the error that we were getting of the fit. So from there, finally, we combined all of these error functions into a vector and then just found the vector norm to minimize that value for a best fit configuration to the animal data. So that was kind of how we did everything. First, then I'll move on to actually presenting the data. First, for the middle legs. So this whole figure is mostly focused on the error positionally between each of the points that we tracked from the animal data and the simulated data. So I'll first begin with these 3 cases. They're kind of the baseline. First, we looked at an all joints mobile case on the far left there that was basically just the baseline with all 7 degrees of freedom mobile, and just to establish how well of a fit we could achieve to the animal data. And the results end up being pretty good overall, especially towards the end of the leg chain. It's important to note that all of these errors are normalized to the proximal segment. So this is in terms of proximal segments. So the reason that this has a slight error here is just because these segments are very small as you get closer and closer to the thorax. The cox and the trochanter specifically are very short. So it inflates the error somewhat, and it also becomes much harder to accurately pinpoint where each of the joint locations is when you get that close to the body, just given limitations to current data capture. So that's a little bit of why there's a bit of error there. But even then, almost like 0.05 of a coxal length is not all that much in the grand scheme of things. We then fixed the THC1, THC2, and TRF2 and found that it did inflate the error in the closer to the body points a little bit, but kept the error towards the end of the leg chain largely the same and pretty small overall. So that was great. We could kind of remove those 3 degrees of freedom, but then we looked into the THC3 and the TRF1, both of which provide a pronation supination motion in the legs. Those are these 2 cases at the bottom here. We found firstly, when we fixed the THC3, and that would be fully fixing the THC itself, we found that we ended up getting higher error in the FTI and tibia tarsus. There's an example of a high positional error frame at the bottom there, and that kind of shows what's going on during that position of error, and that both of those points are not able to really achieve the same level of pronation or supination as the animal in blue. However, the kind of angling of the tibia is roughly the same. And furthermore, as you can see in the ZY projection of the tibia tarsus point over the course of the trial, the limb is still kind of able to achieve the same motions as the animal leg, albeit with just a little bit less movement on either end. By contrast, looking at when the TRF1 is fixed while the THC3 is kept mobile, we find that there is decreased error in the FTI point. However, there is a sharply increased error in the tibia tarsus point. Again, the frame of high error gives us kind of an insight onto why that's happening. And we see that the femur tibia point does end up being much closer to the animal data. However, the limb doesn't seem to be able to achieve the same angling of the leg as the animal data, so that the amount of pronation supination it's able to achieve is much less. To further elucidate the differences and like characterize the differences between these 2 cases, we then looked at the angle of the leg plane, which was the term for the plane containing both the femur and the tibia, to the vertical. And we found that, similar to before, the ranges of the leg plane over the course of the trial, as well as the errors to the animal data, remain quite similar to that of the animal. Whereas for the 2 cases that we were most interested in, we find that, again, the error becomes highest when the TRF1 is fixed. And we find that the reason for that is that the range of leg plane angles becomes sharply decreased without that TRF1 motion, and ends up being far lower than that of the animal, which you can then see here with typical angles over one trial. The limb is really not able to do much pronation supination at all without the TRF1 mobile. Then we'll look at the hind legs. These have some similarities to the middle, but do have a little more of a complicated story. Similar to the middle limbs, we found that fixing the THC1, the THC2, and the TRF2 resulted in minimal errors overall, especially close to the end of the leg chain. When we looked at fixing the THC3 and the TRF1, however, we found that the differences were much less striking as in the middle limb. You can see between these 2 plots that the results end up being fairly similar in their motion capabilities with the tibia tarsus, and the overall error is roughly similar. And we found even that fixing the TRF1 in opposition to what we did for the middle limb, in fact, gave us the lowest error. Again, we wanted to characterize why exactly that might be happening by looking at the leg planes. And we found that, similar to the positional error data that the lowest error occurred when the THC3 was mobile. And then we found the reason for that seems to be that in contrast to the middle limbs, the hind limbs undergo a much less degree of overall angling throughout the course of walking, so that when the THC3 is fixed, that decrease in overall leg plane angle is less of an error than the increase given when the TRF1 is allowed to be mobile. And you can again see that in a typical leg plane angle over the trial, that there's a lot more overshoot with the TRF1 mobile because the overall leg plane range is much lower. So where does this all leave us for robot design specifically? First of all, for both limbs, just because we saw different similarities, we found that the THC1, THC2, and TRF2 could be safely omitted from the overall leg design because they had minimal impact on the overall biomimicry of the system. Additionally, for the middle limbs, it felt fairly straightforward to conclude that the most animal-like joint spatial positions and leg planes occurred when we had the CTR, the TRF1, and the FTI as the 3 degrees of freedom in the limb. So contrasting with the figure that I showed previously, this is a little bit of a mock-up of how this might look using the average angles of those joints that we're fixing over time to create the kind of new zero configuration for these 3 degrees of freedom. The hind limbs are significantly less clear. So there's a couple different things we wanted to consider here. If we were going for strict biomimicry, we would want to go with the THC3 mobile instead of the TRF1. However, we were considering that increased joint planes might have unique benefits to our robotic platform that would allow it to perform better. That might be worth sacrificing a small bit of biomimicry for, given that they were roughly the same in terms of error. In specific, increasing the joint plane, when the body is standing on the ground, there will be a vertical reaction force going up into the limbs. And this force will either be passively absorbed and resisted by the structure of the animal if the torques generated by these forces don't align with the joint axes, or they'll be absorbed by the structure of the or they'll be absorbed by the actuator themselves to hold up the weight if it is torques that are generated in parallel to the joint axis. So by minimizing the amount of that force that ends up parallel to the joint axis, we're able to better distribute the forces and torques throughout the actuators in the structure, and maybe result in using smaller actuators that might be a little more low power and lightweight to reduce the weight of the robot overall. Additionally, it's unclear what the effects of walking on the curved surface are doing to our data, specifically in the hind limbs. I think it's important to point out that the tibia tarsus paths that I showed in the previous figures, for the hind limbs, they're about twice as steep as those of the middle limbs. So there's a great deal of extension downward, down the surface of the ball as the fly is moving. So it could be plausible that the hind limbs would exhibit behavior more similar to the middle limbs if they were walking on a flat surface. So due to these kind of considerations, we elected to also select the same degree of freedoms as the middle, the CTR, TRF1, and the FTI as our final 3 degrees of freedom. So from here, the future steps for the robot design will be to apply this analysis to the fly's front limbs, which those limbs undergo significantly more complicated motion through walking, which is why we kind of neglected to do them in this work because there was limited space. We may require greater than 3 degrees of freedom for these limbs, so that will be the next step. We also want to conduct dynamic analysis of the proposed robot design through simulation. That allows us to test movements corresponding to backward and curve walking in lieu of the motion capture, which is harder to acquire than just running a simulation. This is some very early clips of the simulated robot walking in a forward flat ground. Just to show I'm pretty excited about it. I think it looks very biomimetic to the target organism. And then we'll use the simulation to inform the final mechatronic design. And then I think I'm running a little bit late, so I'll just gloss over these first. Basically, we're going to expand our work with the synthetic nervous systems on this robot, explore companiforms and Scylla discharge, and then apply compliant biomimetic feet to look at the ground reaction forces that those might, how that might alter them. So I'd like to thank the lovely folks at the Neuromint, as well as the Bushkis Lab at the University of Cologne, as well as the C3 and SNRNX group for funding. And I am happy to take all your questions.
Thank you very much, Clarissa, for a very interesting talk. We have time for one question, maybe, or two. Yeah, Nicholas.
I was just applauding. I mean, great job, Clarissa. I don't have any questions though.
Okay, so let's go now with Hillel or Alex.
I was also just applauding. Alex, question.
Great talk. And I was wondering if there might be a way to, in your analysis, build in kind of passive degrees of freedom and those effects. So if you had springs at the joints rather than completely fixing them in place, and how you might anticipate that might affect your analysis.
Yeah, that's a great point. I definitely want to look into that further. Part of what we're doing right now is, in the past robot, we were looking at just passive compliance in the joints and like parallel elasticity to produce some of the same compliance effects as the insect limbs. So we'll be including some of that in the simulation just to add like the spring forces and different damping forces that might come from adding parallel elastic elements. But it would be also very interesting to look at passive degrees of freedom as well and see how that might affect things.
Okay, thank you, Clarissa. Let's move forward now to our next talk of the afternoon, and is presented by Sol Schaefer. I hope I pronounced it correctly. The title of the talk is a computational approach for contactless muscle force and strain estimations in distributed actuation by hybrid mesh constructs.
Okay. So I just want to make sure people can hear me. Is that right?
Yes, we can hear you.
I think. Let's do extend. Okay. Can everybody hear me all right? I think maybe the audio is coming through the wrong place.
No, we can.
All right, I see a thumbs up, so that's good enough for me. Okay, great. Screen is sharing correctly. All looks good to me. So hello everybody, my name is Sol Schaefer, and today I'll be presenting my work on a computational approach for a contactless muscle force and strain estimation in distributed actuation biohybrid mesh constructs. And so I'm presenting on behalf of myself and my co-authors, all at Carnegie Mellon University. So this title is a total mouthful. Let's sort of break down the different components. So distributed actuation robots show this really great promise. And these are just those robots whose actuators are distributed over the bulk of their structure. And in doing so, having this architecture, they're able to change their shape in order to locomote and promote peristalsis internally. And this has great sort of use cases for navigating tightly packed volumes with applications in pipe inspection, in search and rescue, and of particular interest to me in medical devices. And so if we want to make really small sort of mesh-based distributed actuation robots, we seem to run into this roadblock where traditional robots can't seem to be scaled down too far. And that's because conventional actuators seem to have the size limit. Where if you look at sort of the swath of synthetic actuation modalities that are available to us, below around 10 cubic millimeters, there aren't a ton of options. But if we extend our search to the biohybrid regime, all of a sudden we can start working with much smaller materials like single skeletal myotubes or cardiomyocytes, things of that nature. And so the idea here is that biohybrid actuators can permit smaller robots. And so beyond sort of creating smaller robots, they can also drive biohybrid, or these muscle actuators can drive biohybrid robots. And so demonstrated in the literature are these sort of pick and place manipulators. We also see walkers, and we also see swimmers. And all of these can be paced either electrically or optically. And so beyond sort of recapitulating modalities or sort of functionalities that we see in synthetic robots, these biohybrid systems can endow robots with these new, really desirable and unique functionalities. We see properties like self-healing, we see compliance, we see biocompatibility. And of quite interest to me, or great interest to me, is this adaptability, this really unique property of muscle actuators to strengthen with use. Now, as it happens, this muscle adaptability is a double-edged sword. And that's because the relationship between the exercise that a muscle experiences and then the corresponding increase in strength that results from that exercise is poorly understood. In order to understand that better, we need tools for estimating muscle strain to be able to measure that strain that the muscle experiences when it's exercising, and also to know the force that that muscle was producing before it was subjected to this exercise, as well as after, so we can quantify the effect of that strain. And so this lack of design and analysis tools for complex biohybrid systems has led to the state of the art being these rather low degree of freedom lab tethered research toys. And so we have these simple biohybrid systems, but we want complex ones. And that's because distributed actuation biohybrid robots, these more complex biohybrid robots, have these really great potential, right? We can imagine having these trainable, behaviorally flexible biocompatible systems that could adapt to dynamic environments in vivo with long-term applications in medical robotics. We can imagine having stents and endoscopes and minimally invasive surgical tools that could be deployed with these architectures. And so one possible architecture is this mesh-based architecture, where we can imagine having actuators distributed over the bulk of this body. And so while this is sort of something in development and will take a long time to achieve, in the short term, having a mesh-based biohybrid architecture is a very useful platform to study this problem of muscle adaptability so that we can build towards greater complexity in biohybrid robots. So the motivation for this talk is that we have this need for tools to study and design distributed biohybrid actuation systems. Specifically, we need tools that can estimate muscle strain and also for estimating muscle forces in these systems. And we want these tools to meet a couple of criteria. Namely, we want them to be accurate, which isn't surprising. We also want them to be computationally inexpensive, so they run in sort of a reasonable amount of time. And we also, very critically, need them not to introduce confounding variables. That is, that we need them to be quite contactless. Because if we have our sensors interfacing mechanically with our system, they can introduce a mechanical stimulus and then change the way that the actuators will develop. And so the roadmap for this talk is I'll introduce some methods that we developed for estimating strain on a stretched mesh or a stretched substrate. I'll also talk about two methods that we developed for estimating forces on a fixed substrate. The first being a response service optimization method, and the second being a direct optimization method. And I'll compare those two. And lastly, I'll talk about some work that's currently in progress towards physical validation of these computational tools. So let's consider our system as this biohybrid mesh system, right? And let's look at the 2D case. We've got this mesh substrate upon which we're going to pattern these muscles here represented as springs. And what we want to do is subject this whole construct to some stretching exercise regime. We can imagine fixing the top and the bottom and then loading the sides like so. And so what we see is that these the actuators patterned on this mesh experience some strain resulting from the mesh being stretched. And we want to be able to measure that strain. And similarly, we can imagine fixing all edges of this mesh, having the muscles contract, and then the mesh deforming. And in this regime, we want to be able to measure the forces that these muscles are producing. So in order to do this, we developed this finite element model of the muscles patterned on a mesh construct. We have this mesh substrate, which has the following material properties loosely based on a linearized collagen material model. We also pattern these virtual strain gauges, which basically act as these very, very soft springs that allow us to sort of track deformation of different areas of the mesh. And we also have these muscle actuators that allow us to apply specific loads at specific locations, acting as like a surrogate muscle on this mesh. And so let's discuss how we went about estimating muscle actuator strain on a stretched mesh. And so critically here, what's important to consider is what do the muscles actually experience when they're on a stretched mesh? And sort of something that's not quite intuitive is that it's not the same thing that the mesh experience when the mesh is stretched. And that's because the muscle stretch is dependent on the pattern orientation of the muscle with respect to the direction of the stretch of the mesh, right? Is it aligned with the stretch? Is it perpendicular with the stretch? As well as the placement of the muscle on the substrate. Is it near the center of the substrate? Is it near the edge of the substrate? And so when we're talking about how we want to do this, we can consider the one strain gauge situation where we sort of look at a smaller version of this problem, and then we sort of extend it further. So we have our mesh substrate again. We have this virtual muscle strain gauge that allows us to understand the kind of strain a muscle patterned in this location might experience. And then we have what we're going to call the nodes, the places where this muscle would connect to the mesh when it's being stretched. And what we want to know is, given all of the possible locations where we could put a muscle on this mesh, what kind of strains might it experience? And so we can do that by tracking the nodes for all locations where a muscle might be placed, both in terms of position and for orientation. And so what we find when we do this is a rather intuitive result, which is when we stretch our mesh with a muscle patterned, aligned in the direction of that stretch, and then we compress the mesh perpendicular to that direction, this is the sort of loading condition that results in the highest strain, right? And similarly, we can generate a heat map for all sort of possible stretching and compressing combinations to understand sort of the space in which we're operating. And that way we can prescribe a specific strain to our mesh, or rather to our muscle, depending on its location and depending on how we want to stretch that. So equipped with this tool for estimating muscle strain, we now want to look towards estimating muscle forces so we can understand the effect that this strain might have on an in vivo system later on. And so we're going to talk about these force estimation tools for distributed muscle actuators. And so for distributed muscle force estimation, the approach that we took is to fix the edges of this mesh and then load different areas of this mesh with these preloaded springs. And the idea here is we want to observe the deformation of the mesh solely under the action of the muscles that are patterned on it, and relate this deformation to the distributed muscle forces. So we're basically solving this like inverse problem, where we want to be able to measure the deformation of these different nodes and then feed that into a computational tool that will tell us, by sort of backing out, the forces that produce that mesh deformation in the first place. So the first approach that we took to try to solve this reverse or this inverse problem is what we're going to call the response surface optimization method, or force estimation method. And so again, what we're trying to do is take our observed mesh deformation, plug it into our optimization tool, and then spit out muscle force estimations. And so how do we go about doing that? Well, we want to create a mapping between our force space to our mesh deformation space. And we can do that by generating a large set of input muscle forces and then solving the forward FEA problem, and then having a discrete relationship between mesh deformation and muscle force, and then creating a response surface from that relationship to create a continuous mapping of this on this response surface S. And it's on this response surface that we're going to perform the optimization by generating these muscle force validation sets, performing the finite element analysis, getting out our mesh deformations, and feeding those mesh deformations into an optimization problem where we can try to figure out what the muscle force set that was needed in order to generate that mesh deformation. And so once we solve that optimization problem, we can get out our muscle force estimations, and then we can take the absolute value of the difference between our estimation and our validation set in order to quantify our method error. So doing all of this, how did we perform? Well, generating a response surface from 200 muscle force input sets and checking it on 10 muscle force validation sets for a force range of between 0 and around 8 millinewtons, what we find is we don't do quite well enough, right? Our average error is around 321 micronewtons, and our maximum error, or micronewtons, and our maximum error is around 1 millinewton. And so when I was talking earlier about our criteria of having an accurate force estimation tool, this method, while relatively quick at only sort of taking about a minute to perform these estimations, is not accurate enough. So this motivated us to look into other force estimation tools that would enable us to meet our criteria of accuracy. And so that brings us to our direct optimization force prediction method. So with this method, we don't generate a response surface. What we're trying to do here is generate some muscle forces, get out the deformations that are resulting from those forces, and then try to guess, sort of just based on the mesh deformation, what those muscle forces were by performing the finite element problem, or solving it iteratively for better and better guesses for what those muscle forces were, and then converging on a solution, and then seeing how good it was compared to our validation set by taking, again, the absolute value of the difference between our estimation and our validation forces. And so when we perform it with sort of using this method, what we find is that we get an error of 0. And this isn't entirely surprising, right? Because we generated our mesh deformations using the same finite element sort of treatment of our system. And then we use the same finite element model in order to figure out what those forces were. So we'd expected a physical system to see a little bit more error. But what this tells us is that this method of direct optimization has legs, and it's worth trying to validate experimentally. So the trade-offs between these two methods just to sort of summarize this section, is for our response surface optimization method, we have to generate a response surface, which takes days in order to generate enough data to get that response surface. And for our direct optimization method, no training is needed. For our muscle force estimation sets, in terms of the timing it takes for response surface optimization, it's on the order of about a minute. And for direct optimization, each estimation set takes around 11 minutes. So it's significantly longer. And in terms of accuracy, what we're finding for response surface estimation is it's rather lacking in terms of its accuracy. And for direct optimization, it's quite accurate in silico. It remains to be validated experimentally, which sort of motivates the work that we're doing right now that's in progress, which is towards validation of the experimental tools that of the estimation tools that we've developed, because simulations are doomed to succeed, right? We need to ground our work physically to make sure that it's actually useful to us. And so we have some requirements for our physical validation, right? We need our system to sort of recapitulate what we've created in silico. And so we've got these two components, right? We've got our surrogate mesh, sort of seen here in that white elastomer. And we also have a fiducials sort of placed on it. And we're trying to track the relative displacement of these nodes, of these fiducials, optically, so we can understand how this mesh is deforming, both based under the action of the biaxial stretcher on which it's placed, as well as the muscles that are pandered on it. And so yes, we have these muscle springs and they're pandered on our mesh, and they can apply known loads to our system. And we also have this stretching rig that can allow us to apply known displacements to the system, and thus we can do the tracking and try to validate our work. And so this is in progress at the moment. So in summary, the technical contributions of this work are a contactless method for estimating muscle strains on a stretched mesh, as well as two contactless methods for estimating muscle force. One is quite fast, but rather imprecise, so not entirely of use, and one is rather slow, but quite precise. So this is the one that we're going to sort of push forward on moving forward. I'd like to acknowledge the funding sources for this work, and also the people in my lab who have been fantastic resources to me, especially the undergrads who have worked under me on this project. Thank you so much, and I'd love to answer any of your questions.
Thank you, Sol, for this fantastic talk. Does anyone have any question?
We seem to maybe have an audio problem.
So
I don't know if I can hear you.
Well, I can hear you.
We do a little
Saul,
switch.
can you hear me? Saul, can you hear me?
Yeah, go on, Hillary.
I'm just checking whether Saul can hear me. Saul, can you hear me?
Okay, now I can hear you. Yes, please go.
Okay, just a quick question. Is there any way of combining the two, since they each have sort of, what should we say, complementary drawbacks? In other words, the rough surface is not very accurate, but it might give you a ballpark estimate that would allow you to do the FEA online much more quickly. Is that possible? Would that be useful?
Yeah, I believe it would be possible, right? Where you can have like an ensemble method that combines sort of the best of both worlds. That's something that I'm working on. Yeah, great suggestion. Thank you.
Sure.
Great. I think we have another question by Bidisha.
Yeah. Hi, Sol. So,
Hi.
yeah. So, I have a little bit confusion. You may have mentioned it that your optimization is constraint, or is there any constraint, or it is not without any constraint? And in your slide, I forgot the slide number. There are two functions, FV or FP. So, may I know which variables present in their argument of this function? Means I can understand that they are not function of same variable, okay, because one is muscle force and another is, I think, your not things force. So can you please elaborate?
Yeah,
thing?
I'll answer
Yeah.
your second question first. So we have our muscle force validation sets, F sub V. And so this corresponds to 4 muscle forces. Those muscle forces in particular are these muscle forces. So they correspond to the forces on these different muscles. So this is muscle A, this is muscle, oopsie, you can't see my mouse. This is muscle A.
Yeah, I can see.
Muscle B, this is muscle C, and this is muscle D. So it's just, it's not a function, it's more of like a vector that houses 4 muscle forces, if that answers your question.
Yeah, so may I know that with respect to which variable you are trying to optimize?
Sure,
means
yeah. So what we're trying to do is we have,
yeah.
let me just navigate to the correct slide. What we're trying to do is we're trying to use mesh deformation as an input and then find the muscle forces that most closely match those mesh deformations. That's sort of our goal with our optimization. So our goal is to try to find the muscle forces that correspond to a mesh deformation that are most closely resulting in the mesh deformation that is the input based on the response surface for the response surface optimization method. And for the direct optimization method, it is solve the FEA problem with your best guesses of the forces and minimize the difference between the mesh deformation that results from that solving and then the input mesh deformation. I think you might give it a in the paper, and so maybe if you take a look there, it could be like a slightly more
yeah.
better.
Yeah, okay, because we don't have more time, unfortunately.
Okay.
But let's see if we have time at the end, like for the rest of questions, because I saw there is one more. But for now, we need to continue to our next talk and next and final talk, which is by Wei Huang. And it's the title is Surrogate Modeling for Optimizing the Wing Design of the Hawk Moth Inspired Flapping Wing Micro Air Vehicle.
Hi, thank you for the introduction. Let me share my screen real quick. All right, can everyone see my slides?
Yes.
Yes.
Okay, so yeah, I'd like to thank the conference committee first for giving me the opportunity to present my research, and thank you everyone for being here. So the work I'm presenting today, it's done for my master's thesis research at the Biologically Inspired Robotics Lab at CASE here. So in this work, we're investigating the circuit modeling approach as a method for essentially rapidly estimating computational results in flapping wing aerodynamic simulations, and then using that circuit model to determine optimal wing design solutions for a flapping wing micro air vehicle. So a quick overview of the presentation. I'm going to go through some of the current design challenges in developing FWMABs and the contributions from our circuit modeling approach that addresses these challenges. I'll talk about the method I used to model the four-wing geometry and flapping kinematics for the moth in hovering flight, the simulated results in the CFD simulation that study the aerodynamics around the wing. And then finally, I'll talk about the support vector regression circuit model that was trained and its performance in predicting the results from the CFD simulation. So most engineering design concepts usually go through an iterative design cycle that involves multiple experimental analyses using both high fidelity computational simulations or real world experiments for design validations. So in the case for flapping wing aerodynamics, you usually use CFD simulations or wind tunnel testing. So this design process can involve numerous computational expensive experiments when we're trying to solve problems like determining the optimal design for a system. So this can be especially challenging when you're dealing with bio-inspired systems that has a complex system architect because it's based off of biology. So in the work here, we looked at the principle behind the effects of the moth wing shape on the aerodynamic performance in hovering flight. We studied this relation by designing engineered wing models with variable wing geometry parameters rather than studying just one wing design that directly mimics the hawk moth. So when we're studying the aerodynamics of the flapping wing in hovering flight, digital modeling of the wing, performance in computational simulations can become increasingly complex as we start to consider the unsteady aerodynamics around the wing, the flexibility of the wing as it interacts with the unsteady fluid, and then the variation in wing motion and geometry and how that affects the aerodynamic behavior around it. So with all that considered, computational simulations used to investigate flapping wing aerodynamics can become increasingly expensive in terms of both time and computational demand as higher fidelity is built into the model. So to address this, I investigated the circuit modeling approach as a method for estimating the results of these expensive to run computer simulations in a quick manner without the need to run multiple iterations of simulations. So the circuit models are essentially statistical models trained on the data gathered from real experiments or CFD simulations. They are used to accelerate the engineering design process. Essentially, circuit models are capable of estimating the output from real experiments to allow for rapid model sensitivity analysis, solution to optimal design configurations, or rapid design space exploration. So in this work, we designed an experiment to investigate the variation in the wing geometry parameters of engineered hawk moth wing as it relates to the aerodynamic performance of the wing in a CFD simulation. Then the results of the experiments are used as training data for supervised machine learning model to rapidly predict the lift to drag ratio of all possible wing geometry configurations in the design space of the wing. So for modeling the 3D wing, I considered essentially 5 design parameters: the wingspan, the maximum chord length, max camber as a percentage of the chord, so that's M, and then max position, the location of that max camber P along the chord length, and then the max thickness of the wing T. So the bounds of this design space, it's defined as the minimum and maximum values in a sample set of biological wings that was collected in O'Hara's paper in 2012. So after that, I use a Latin hypercube sampling method to draw essentially 20 samples of wing designs in the design space. So in figure 4 on the right here, you can see the design space samples in 2D space for every combination of pair parameters here. So by using the 2D forwing of the moth's 2D image, by using a 2D image of the moth's forwing, we can scale essentially a 2D point cloud representation of the wing accordingly to what our desired wingspan and chord lengths are. And after the wings are scaled, we can project that into a mean camber surface function that was also measured in O'Hara's paper. So I just projected my 2D wing design onto the mean camber function here. And then for the cross-sectional geometry of the wing, I modeled the cross-section as essentially NACA 4-digit airfoils. The design parameters M, P, and T here, they're the maximum camber, the position of that max camber, and the local thickness of the wing along the span. So with this model, we can vary the camber and local thickness of the wing section within the bounds of our design space, right? So you can see here the thickness of the wing along the wingspan, it varies from the root to the wingtip. So I set a variable thickness parameter here with the thickest part of the wing being at the root, and then the thinnest part of the wing is located essentially at the tip of the wing. So with all 5 of the wing geometry parameters defined, we can create a fully 3D geometric model of the engineered wing. The third image here in the figure, it shows how the thickness along the wing is distributed based on the parameters that I've defined here. So after we define the wing geometry, we can go into deriving the kinematic equations for the wing motion. So the model for the kinematic variables I use here, it was first developed in Wilmot's paper in 1997. We're defining the body angle with respect to a fixed inertial frame, and then the stroke plane angle, elevation angle within that stroke plane, and then the sweep angle in the stroke plane. With the maximum sweep angle is defined as essentially the pronation period of the wing, and then minimum, that's when supination occurs. Okay, So with using Wilmot's measurements for those kinematic equations, I used essentially a coordinate transform, 3 coordinate transform matrices from the body frame of the wing to the fixed frame to get a motion for the flapping kinematics. So we can see here, this is for hovering flight at a constant stroke plane angle of 23.4. So in my models, I'm assuming the stroke plane angles are constant. But in Wilmot's paper, this angle actually increases as the floor flight velocity increases. And then your rotational angle actually decreases a little for higher floor flight velocities. So with the 3D motion of the wing derived, I'm looking at just one 2D section at 70% along 70% along the wingspan. That cross section specifically, I'm putting it into a 2D CFD simulation. So first, before we go into the CFD simulation, in the 2D kinematic equations in the fixed frame, we can determine both the airflow velocity magnitude and essentially the effective angle of attack of the incoming air acting on the wing body in the wing body frame. So because there is no incoming air from hovering flight, the effective airspeed is just the magnitude of the wing velocity in the fixed frame. And the effective angle of attack is just a function of wing rotational angle and the components of the wing velocity in that fixed frame. So from that, we can look at the effective airflow velocity plot and can observe the wing has essentially a minimum airflow velocity at mid-supination, and then the max airspeed occurs essentially during upstroke. And then looking at the effective angle of attack transitions from negative during upstroke to positive during downstroke. So to calculate the fluid flow regime, the Reynolds number, I used the average airspeed and then the average chord length in the 20 samples. We calculated a Reynolds number of around 5,600 for hovering flight. So looking into the CFD simulations, we used a pressure-based solver in ANSYS Fluent. The governing equations used are the 2D incompressible Navier-Stokes equation for unsteady flow. So we imported the 2D wing geometries in the design space samples into ANSYS, and then defined the airfoil as no slip boundary condition at the wall. And we're running transient simulations for all wing geometries using dynamic meshing around the wing. So the dynamic meshing functions are defined by the kinematic equations that we derived before in the previous section. I also performed a grid sensitivity analysis for the CFD simulation to reduce the runtime in the simulation. So after testing all the grid resolutions, the medium mesh and the fine mesh, they had results that agreed with each other, but the coarse mesh had differing results. So we use the medium mesh for all simulations just to reduce the runtime. So here's the simulation of the 2D wing conducted for 4 flapping periods. So the airfoil and fluid initially start at rest. The velocity contour in the simulation shows essentially the leading edge vortices being formed during downstroke and upstroke, and then instances during the upstroke phase where the wing tries to capture the wake that was formed during downstroke. So the 3 lifting mechanisms that we can see here, there's the delay stall occurring during downstroke that forms the leading edge vortices, and then during that transition period between downstroke and upstroke, the wing goes into generating rotational lift. So rotational lift, that's essentially producing lift using the Magnus effect. And then after that, the wing essentially goes into upstroke to capture the wake. So for the results in the simulation, we're calculating the 2D forces as components of the pressure and viscous forces in the simulation, and then using a approximated 3D force calculation, we're just multiplying the 2D forces by 2 times the wingspan to calculate the total force for both wings. And then from the 2D forces, we're calculating a lift to drag ratio just by dividing the two time average forces. So for all the samples, the figure you see here are the total approximated 3D forces for each wing sample, and their average lift to drag ratios range from highest to lowest. So what we found was actually wing sample 11 here had the greatest lift to drag ratio out of all of them. Interestingly enough, this wing had a cross-sectional geometry that most resembled that of the hawk moths. And then in the circuit modeling part, once the lift and drag ratio for each sample are calculated, we're using the training data set for supervised machine learning with the predictor variable defined as the wing geometry parameters, and then the response variable are the lift to drag ratio calculated from the simulation. So I used a support vector regression model using 3 different kernel map functions on the training data set, and then they're trained to minimize essentially the root mean squared error of the function. And after training the SVR models with all linear, quadratic, and cubic kernel functions, the model with the smallest root mean squared error and predicting the training data set was the quadratic kernel SVR, and the root mean squared error for that and the R squared values are presented here. So using the trained quadratic SVR model, predictions on lift to drag ratio of varying wing designs inside the design space can be made. Each contour tile here shows the variation in lift to drag as a function of 2 wing geometry parameters, while the other parameters are fixed at the values listed on the right here. Here, the fixed parameters in figure 16 are set as the minimum design space values. And if we transition to the next frame, we set the fixed parameters in the middle of the design space. And then here's at the maximum design space values. So these contours, essentially, they predict CV over CH for all the possible design space configurations. And then if we move on to the conclusion, so from the lift to drag ratio contours, we can determine parameters that have the greatest influence on the lift to drag. So the plots that show a greater contour gradient belong to parameters that vary the lift to drag ratio the most. So the goal of this research is just to show the capabilities of circuit modeling and predicting the results of a computationally expensive CFD simulation that involves complex flapping wing aerodynamics. And therefore, with this finding, we can rapidly find solutions for optimal wing designs doing a gradient search. So ongoing work currently is looking into validating optimal design, wing design with an additional CFD simulation to see how accurately the support vector model predicts the CFD simulation. So future work for this project, I want to focus on building a higher fidelity into the simulation model, which would essentially include expanding the 2D simulation into 3D, because 3D aerodynamic mechanisms such as the wingtip vortices are have to be considered for the wing, and then stuff like the flexible wing model have to be considered for fluid structure interactions. taking the project even further would have to involve moving the experiments into a real world wind tunnel setup with a compatible flapping mechanism. Okay. And I want to thank my advisors, Roger Quinn, Kenneth Moses, and Brian Schmidt for their contribution. Thank you for listening. That concludes my presentation.
Thank you for a great presentation. I think we have time for 1 or 2 questions. If there's any of the in the audience. So I have a small question. Like, so in the end, you show like the results of the surrogate model, but I never saw like the comparison with the actual simulation. Do they match exactly, or like what is the error?
So yeah, that's ongoing work right now. I'm
Oh, yeah,
working on finding what the optimal wing design is, and then testing that in a final CFD simulation to validate it with the SVR model.
And the like these contours, like do they have like a smooth change? Like do they change smoothly as you change the parameters, or do they have like a, I don't know, like some sort of sharp changes for some parameter values?
Yeah, they do change smoothly. So because it is a smooth function based on the SVR model is a smooth function. The mapping I'm using is a quadratic feature.
Cool, great work. Any more questions? Okay, so if not, I think that concludes this session for today. I don't know if Alex or anyone has anything concluding remarks.
Thank you, everyone, for coming out today. We can continue discussions on Discord, and we have one more great day of talks planned for tomorrow.
Thanks, everyone.
Thank you, everyone. Bye.